say, describe the structure of DNA. Structure of DNA. Guys, when talk about the structure of DNA, look at the characteristics of DNA. So I think that's done. The characteristic of DNA is double helix is made up of 2 strands. It's made up of the nucleotide. It's made up of nucleotide, yes, which are made up of phosphate, the oxyribose sugar, and nitrogenous bases. We talk about the nitrogenous bases, talk about all of that. Yes. Challenge. Don't go to the paper without knowing DNA replication. Yes, don't go to the paper without knowing translation. Don't go to the paper without knowing transcription. And then describe the description of the structure of DNA. So one will come. Won't tell you which one, but one will come. You'll come. We're going to discuss the paper after. You'll know. And then now, if they ask you a question that give the difference between DNA and RNA, that one is simple. You can talk about everything you know. Make sure that you draw the table. Don't leave it hanging like this. You want a complete table. You mentioning this, you writing like this, and you say DNA, and the other side you say RNA. Guys, put DNA, put RNA, close the table. If you don't know anything, at least you got a mark. Yes. If they ask you to tabulate, draw a table, put the title, close the table, leave. If you don't know anything, yes. If you know something, then write something. Yes. I'm done with that. But now the challenge comes in when they ask you that give the difference between DNA nucleotide, nucleotide, and RNA nucleotide. Here, you only look at the nucleotide of DNA, the nucleotide of DNA and the nucleotide of RNA. And there are only 2 differences in there. DNA has, don't talk about the phosphate because it's the same. DNA has deoxyribose sugar and the RNA has ribose sugar. And then DNA has thymine, RNA has uracil. Done. If they ask you about what? About the nucleotide. Is it clear? If they ask you the difference between DNA replication and transcription, you have to know the differences. DNA and transcription. We're going to spend a few minutes here. I'm only going to focus on the things which you fail. Which you fail is what I'm going to focus on. Yes, too much. Then we are saying that DNA, you talk about DNA, in this case, in DNA replication, 2 strands act as a template. The other side, you talk about 1 strand transcription, 1 strand act as a template. Guys, only know at least 3, but we want 2. In most cases, we want 2. We won't ask you more than 2, the differences. Don't go to the paper. If there's a lot, just know few things. Don't on each, at least go have a dose on each, whatever I'm going to talk about. Yes, then you'll be able to make it. Then we are saying that, yes. Then another thing which you go off is about the mutations on DNA. About the mutations on DNA. So when they ask you about the mutation, let me just use my book here so that you understand. Excel in Life Science Distinction Material. Here, let me just, I don't want the structures. Okay, I don't want the structures. What I want is this. What I want is at the end here. Yes, at the end, the mutations. Yes. Guys, let's answer this question. That what will happen if a mutation changes triplet number 1? Triplet number 1, it's this. From this, the same thing. From this to this, it means that this has been changed. Is it clear? You changed G to A. So now what you do when they ask you such a question, you first identify what is here. It is a codon. What is given here is DNA. Therefore, I have to convert this into messenger. Remember, we say that. Let me do like this. Yeah. Remember, we said that when you talk about code, it means DNA. DNA. When you talk about codon, it means messenger RNA. 3 bases on messenger RNA. When we talk about anticodon, we mean 3 bases on transfer RNA. So now the question is asking, it means that it's changing from DNA to codon. It means that it's talking about messenger RNA. Is it clear? Yes. So now, what you do is to change this into messenger RNA. So change this into messenger RNA. So when you change into messenger RNA, this is going to be A goes with U, T goes with A, G goes with what? G goes with the C. So I'm going to CUA. I come here, look for C, C, CUA, C, no, AUC, AUC, where is it? US is triosine. So this is the triosine. UCA, UCA, isn't it? Then what about this? Now I change it again. Also is U. It's gonna be. It's gonna UAU. What does it mean? UAU is also triosine. So it means that a change from this to this, yes, will lead to this USC, from USC to UAU. That's the first mark. It will, you converting this into codons. You understand? Into codons. Is it clear? Which will code for the same amino acid, triosine. Therefore, there will be no change in the protein. Why? Because it's the same amino acid which is being coded for. Can I repeat it? I said, when they bring such a question, I know when they bring it, and I know it will be there. When they bring such a question, students lose these marks. Change in this, first find out, changing, because here it might be tRNA, and here is codon. So you change into the codon, which is messenger RNA. Is it clear? So change from ATG, basically result in this, and the change in this result into this. Is it clear? And these two, they code for the same amino acid. Therefore, it does not code for a different protein. It's like me calling you, let me check. Let me check here. It's like me calling you Lerato, Lo, Lailo, whatever. Ne? Sorry if I pronounced you wrongly. Ne? I can call you Lerato, or I can call you whatever, ne? The second name. I don't want to pronounce it badly. Ne? Yes. So if I call you the other way around, you say yes, sir. When I call you with another name, you also say yes, sir. You understand? So it means that they are coding for the same amino acid. However much the codes are different. Is it clear there? Yes. Then, what about base triplet number 4 from, yes, base triplet number 4. So this is the first instance. The second instance, what will happen to a protein if the base triplet number 4, ATA, is changed to ATT? So ATA, I have to convert it because the DNA, I need them to be in messenger RNA. ATA is going to be, T goes with A, U. And then this one is going to be U. So now I say that this will change into this. Okay, this will code for this, and this will code for this. Cool. That's point number 1. But what does this code for? AAU. AAU is valine. What about UUU? UUU, is it valine? No. Where is it? We have UUU. No, man. It's not UU. Yeah. What am I doing? It's AAA, ne? It's AAA because T goes with A, T goes with A. So it's AAA. So what am I looking for? Is the AA is proline. So it means that if you look here, this one code for valine. While this one code for proline. You see? So it means that these two, they are different. Is it clear? So it means that even the protein will change. It's like calling you Carabo, and then I call another, I call again Cabero. And Cabero is different from Carabo. You see? Yes. So it means that now this will code for this will code for this, of which this code is for valine, and then this, you write this in full, that AAA, ne, which code for proline. Therefore, a different protein will be formed because of the different amino acid. That's the scenario number 2. Scenario number 3. Yes, one scenario will come. Ne? Explain what will happen to a protein if DNA base triplet, or if DNA base has changed. Yes, there they have not talked about you have to interpret it automatically. If the DNA sequence has changed, one base has removed or has been replaced, whatever, it will lead to a different sequence of messenger, which will lead to a different sequence of transferrin A. Yes, which means a different sequence of amino acid. You understand? Hence a different protein. Different sequence of DNA will lead to a different sequence of messenger RNA, which leads to a different sequence of transferrin A with its amino acids, which leads to a different protein. That's a study scenario. The first scenario, it might not change the protein. Therefore, the protein is not going to change because the amino acids are being coded by different codes as we saw it. The second scenario, they can code for a different amino acid, meaning that valine and proline, for example, therefore a different protein. Or they can ask you what will happen if DNA base sequence changes or messenger RNA sequence changes. So you start from messenger RNA sequence. If it changes, it will lead to a different sequence of transferrin A with its amino acid, hence a different protein. Guys, don't go without knowing those things. Lastly, on DNA, it's this. Let me just use this example, but you still have questions. You still have questions we can use. Please, 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 don't go to the paper without knowing that. Someone is telling me they have given you T or C. Just mind A and C. Yeah. So guys, make sure that you guys get the things right. Yes. If you have any challenge, let me know. Yeah. Lucy. Lucia. Yes, you're saying adenine join. Yeah, I'd made a small mistake, but yeah, I saw it when I was trying to. Yeah, I'm also human. I can make a mistake. Yeah, but when I do it, sometimes when you talk, you can talk about something else, and then what in mind is something different. Yeah. All right, but I'm sober today. I'm sober. I don't drink and smoke. So I'm sober. All right. All right. Let's continue to the next. Guys, don't go without knowing how to label these things. What is this? It is amino acid. What is this? Is ribosome. What is this? What is this? It's transfer. You see, it is a T. Guys, check. It's a T. tRNA. If you don't know it, try to see if that T will come up. Check. You see? It comes out. It's a T. So it's a T transferrin. Guys, this is not a protein. It's a polypeptide chain. Protein is 50 amino acid and above. Is it clear? So this is DNA molecule. This is messenger RNA. This is nuclear pore. This is the nuclear membrane. Please go when you know how to label these simple things. Is it clear? Yes. This is the transcription. It occurs in the nucleus. This is translation. It occurs in the cytoplasm. Therefore, if they ask you what is this space, is cytoplasm, and what is this space, is the nucleus. Yes. Lastly, but not the least on DNA, so that we go to meiosis. Two things on here. The first thing is if DNA is 30%, if adenine is 30%, what is the ratio of guanine to cytosine? What's the ratio of guanine to thymine? Let me just say like that. Guys, you have to know how to do the base calculation. A goes with the T, C goes with the G. If this is 30%, automatically also this is 30%. So 30 plus 30 gives me 60. So remember, this is percentage. Percentage means 100. So 100 minus 60, it gives me 40%. So this 40% is for these two. So divide by 2, so it's going to be 20, it's going to be 20. That's not the answer. They're saying the ratio. So it's going to be G to T, whereby G, where's G? G is 20 and then T is 30. So I divide by 10, I divide by 10. So I get 2 here and then I get 3 here. You know what you're going to do? The first objective we're going to do, we're going to say that 3 to 2. Because when you saw 3 and 2, you just say that it's A. And the last one is going to be 2 to 3. because you don't want to read everything. Make sure that you read everything because there is most correct answer. I remember in paper 1 whereby they gave a correct answer halfway correct, and there is the most correct answer just down. You understand? We always try to trick you to see that, yeah, you are off. But I know since you are here, you won't be off. Ne? Yeah, we are here to kill the paper, not the paper to kill us. Ne? Yeah, tomorrow we will make it. We will make it. We'll make it. I know. All right. Yeah, if do you have any question, let me know. Let me know if you have any question. Let me know so that I smash that question without. That's number 1. That's number 1. The last thing which I need to talk about is about profiling. This profiling. Someone might say that profiling is easy, man. Yeah, I know that profiling is easy, man. But there is a time when they are talking about comparing the paternity test. Ne? Let me see if this question is about paternity test. Identifying the criminal, or what is this? Okay. Sample, DNA sample. Okay, let me just read it. Detective investigating the scene of broken window, suspected blood was found in the criminal, identified. No, it won't give me what I want. Blood. And it won't give me what I want. Okay, now let me design my question so that now you understand what I'm saying. If they say that 2 guys, they are crying, or they are saying that, okay, you have a boy, and then you have 2 guys, ne? And these guys, they are saying that it's each is saying that is the father of this boy. But now the mother doesn't know who, because this mother slept with these guys, ne? These guys are the same day, ne? You understand? So now we don't know who is the father of this boy. So what you need to do is, if this is the DNA profile of the boy, guys, listen until we end this, ne? Because sometimes this question, when it comes, you are off. Ne? Okay, if this is the profile of the child, yes, if the profile of the child, I know DNA profile is very easy, but once it comes to this, you lose the marks. Yes, you lose the marks. Okay, this is the boy. Ne? And then now you have the profile of the mother. You have the what? The profile of the what? Of the mother. You have the profile of the mother. Is it clear? Yes, you have the profile of the what? Of the mother there. And then now you have the first profile of the guy. Well, the first profile of the guy. You have the first profile of the first guy. You have the first profile of the first guy. Ne? Have the first profile of the first guy. Yes. And then you have the second profile of the second guy. You have the first, you have another profile of the second guy. You have another profile of the second guy. Yes. Yeah. So now they ask you, this is the mother, this is X, this is Y. Is it clear? Who is the father of this guy? And after that, they can ask you to explain, and they say 4 marks. Explain 4 marks. That's it. That's what I'm saying, that's where students go off when it comes to this. Listen carefully what you're supposed to say. Why did I divide with the 10 so that I get the simplest ratio? Ne? Yes, I divided by 10 so that I can get the simplest ratio. Yes. To simplify the ratio, yeah. Yes, correct. What you said is correct. I divided by 10 so that I can get the simplest ratio. Equilibrium, I will reach there, don't worry. So, guys, look here critically. What you need to do here is to first, because I know the mother. So take all the bars which correspond with the mother. So this corresponds with the mother. Yes. This bar corresponds with the mother. Yes. This bar corresponds with the mother. So it means that, let me just use a different color so that we see exactly. So this bar is out. This bar is out. This bar is out. So now the remaining bars, not that one. I'm saying this bar is out with the mother, this bar out with the mother, and then this bar is out with the mother. So the remaining bars, these remaining bars, this, and this, it must correspond with one of the guy. If there is one bar which is not corresponding, then that's not a father. Is it clear? Yes. Now I come here. I look for this. What does this one correspond with? This. If there is no one who is corresponding with it, then it's out. You understand? Then is what? Is out. So this bar correspond with the with this, ne? Yes. So it corresponds with that. This one is a little bit down. And then this bar corresponds with that. This bar corresponds with this and this. So however much this one has one which is corresponding, but this one and this one did not match with it. So the remaining bars, you see that all the remaining bars, yes, have corresponded with this guy X. Therefore, guy X is the father. Explain your answer. Why are you saying that X is the father? X is the father because the bars of the boy correspond, the remaining bars which of the boy which do not correspond with the mother correspond with this guy X. All the remaining, the word all must be there. All the remaining bars of the boy, yes, after matching them with the mother, correspond with guy X. Is it clear? Because they always ask you this. Yes. And then students tend to go off. These bars must correspond with the mother. The remaining bars must correspond with the father. If there's one bar which is not corresponding, we will ask you, where did that boy get that gene? Understand? Remember, we get these chromosomes as a sperm and ovum. Where did that boy get that gene from? Are trying to mean that's a mutation? So that's why all of them must correspond with the father, so that we conclude that that gentleman X is the father of that boy. Yeah, guys, unless you have any question concerning about describe the process of translation according to the messenger RNA, the transfer RNA with the complementary anticodon match according to the messenger RNA, the transfer RNA with the complementary anticodon, yes, bring the required amino acid to the ribosome. The amino acid is joined together by a peptide bond to form a required protein. Guys, those are just processes I'm not going to go through. Maybe I can ask a question. What is DNA replication? When does it occur? When does it occur? When does it occur? It occurs during before the cell divides, during interphase. Where does it occur in the nucleus? Why does it occur to double the chromosome number? Yes. How does it occur? That is the process of DNA replication. So there are so many questions. I'm not going to go through them, but make sure that you know those processes. Unless you have a question about DNA replication, about DNA code of life, let's go to meiosis. Yes, when we talk about meiosis, guys, there are some things which you need to talk about. See, this is a transfer. Okay? It makes it. You see, this is amino acid. This is transfer. We said it. This is a ribosome. This is messenger RNA. Sometimes it's up, sometimes it's down. This is what? Codons. Hey guys, how many? This question. 2 questions. The first one is how many codons in 12 bases. So remember, 3 bases is equal to 1 codon. So it means that 12 divided by 3, so makes them to be 4 codons. How many bases, how many amino acids in the 4 codons? It's gonna be 4, because each codon code for 1 amino acid. Understand? How many bases code for 5 amino acid? Remember, each amino acid is coded by 3 bases. So I'm gonna say 5 times 3, which gives me 15. You understand? So you have to know how to interpret that. And then, let's go to meiosis. Yeah, we try to explain this. They say left, right. Make sure that you translate it left, right. Ne? Yes. The first thing you need to know about meiosis are the phases, but I'm not going to go through the phases. Use IPMAT to be able to know which phase is it. But guys, something is itching me. And that itch makes people to lose marks in meiosis. Number one, identify phase in diagram 2, diagram 1, diagram 3, and diagram 2. Guys, whenever they ask you about the phase, please turn your paper if these centromere, the centrioles, they are not on the sides. So turn your paper to be like, yes, you see, it must be, I don't know, I think this is east and this is west. If I'm not a geographer, I hope this is the correct. Yeah, but it must move east to west, east to west. It must not leave what? East to west. Don't leave this, they must be in east to west so that you identify the perfect what. If it's not straight with a paper, turn it. Turn it, because if you turn it, you will be able to identify the actual what? The actual phase. So in this case, because there is space here, it means that is anaphase. But because it is single, therefore is gonna be anaphase 2. So it's anaphase 2. What about here? The chromosome, you see there in the equator. So it's gonna be metaphase 1. Yes, because the chromosomes are double, you see, therefore it's gonna be metaphase 1. If they're single, then it's gonna be meiosis 2, which is you identify the phase and then you put the word 2. That is the first thing which you need to know before you go for the paper. The second thing which you need to know about before I tell you what to focus more on this is the number of chromosomes. How many chromosomes are found in diagram 1? You will find out someone saying 46. Did they say this is a human cell? No. If they don't talk about human cell, don't talk about 46, don't talk about 23, because they didn't talk about human cell. How many do you see? 1, 2, 3, 4. It's 4. How many chromatids found in diagram 1? It's 1, 2, 3, 4, 5, 6, 7, 8. It's 8 chromatids they're talking about. How many chromosomes will be found at the end of division of diagram 2? Yes, because this is meiosis 1, therefore it's gonna be, since there are 4, therefore it's gonna be 2. Yes, that's the number of chromosomes which will be found at the end of meiosis 1. Now the question comes, how many chromosomes will be found in diagram 2 if this was a human cell? Now they said if this was a human cell. Because this is meiosis 1, human cell, therefore there will be 46, or there will be 23 pairs. Yes, I think I'm clear there. That is point number 1 about meiosis. Point number 2 is about the karyotype. Point number 2 is about the karyotype. This is where also you lose marks. Describe the structure, describe the karyotype. It's fine. You can describe that from this karyotype is for a male. If you see this is for a male, this is for a female. Because here, the chromosome number 23, they have the same size and shape. While this one is big, one is small. Yes, therefore it means one is X, one is Y. But now, if they ask you to describe the karyotype, from is made up of 46 chromosomes, which are arranged in pairs. From pair number 1 to pair number 22, we call them autosomes. The 23rd pair is called the gonosome. The gonosome has X and Y. If it is a male, has X and X. If it is a female, the X is longer than Y. So you describe what you see. But if they ask you how many autosomes are found in P, please guys, don't just say that is 23. Sorry, don't just say that is 40, 44, or is 22 pairs. No. Count them, because sometimes maybe chromosome number 6 we put them 3, whereby you say 1, 2, 3, 4, and you don't see that, you don't recognize. Look at chromosome number 21, but it's better to count them 1, 2, 3, 4, 5, because you'll find out. They won't just ask you a question, how many chromosomes are there? No, man, they will have to hide something. There must be a reason why they ask you that. Yes, there must be a reason why they ask you that. Maybe there is a chromosome which they have they have done the hiding, and then when you have to identify where they have hide that. Guys, give it or don't panic. Take your time, read the question 3 times exactly. If you feel like you don't understand, skip it. Yes. Why? Right? What? Start. If you see that things are not working out, start with what you think is right. I'm telling you, don't panic. Don't panic because you have not seen the diagram, Yeah. The diagram is this time they have brought it in terms of what? So why are you skipping? Skipping what? I'm not skipping anything. Yeah, I'm going to come back. I'm trying to talk about the things. After that, I come back to those questions. Ne? Yes. So guys, make sure that you concentrate on those karyotypes. Make sure that you know how to identify them. Give a reason. Give one observable reason why state the gender of P. P, where is P? This one. We say that this P is what? Is a what? Is a male. Why? Because the chromosome number, the pair number 23, it has 2 different chromosomes. One is X, one is Y, or one is big, one is small, meaning that one is X, one is Y. Yes, that's number 2. That's where people fail. Number 3, non-disjunction. Non-disjunction. You see this? This is a non-disjunction question. Yes. Whereby when they ask you to which phase is it? Is anaphase. It could be anaphase 1 or anaphase 2. Yes, but looking at the diagram, you can identify that it's anaphase 1 or anaphase 2. Yes. Describe Down syndrome. How is it? How is non-disjunction lead to Down syndrome? Guys, you just talk about what is extra. That Down syndrome is as a result of non-disjunction, leading to one cell having an extra chromosome. Yes, on chromosome number 21. Yes, meaning that it's gonna have 24 chromosomes instead of 23. Yes. When this cell fertilizes a normal ovum, the resultant individual is gonna have 47 chromosomes instead of 46, leading to Down syndrome. Is it clear? Down syndrome is caused by non-disjunction. Is it clear? Either in anaphase, it could be anaphase 1 or anaphase 2, leading to a cell having an extra chromosome on chromosome number 21. In brackets, you can say 24 chromosomes instead of 23. Then, when this gamete, yes, fuses a normal gamete, meaning that it is having 23, it will result in 46 chromosome, a zygote with the 46 chromosome, 47 chromosome instead of 46, leading to Down syndrome. It's simple. I don't know why people make it difficult and complex and what. No, man, it's simple. Is it clear? Yes. So that is another thing where you lose marks. Number 3, give 2. Let me check here. I hope that question is here. Metaphase 1 and anaphase 1. Those ones you can do. Okay. Describe 2 ways in which this is Down syndrome. Yes. So we have answered that one. Yes. Name the organ in male that where meiosis occur. Okay. Meiosis occurs in the testes for the case of male, and then female occurs in the ovum, ovary to produce ovum. Yes. But there is something which I want where students fail. Give 2 causes of variation in meiosis. In meiosis. Guys, they are saying in meiosis. In meiosis, they're not talking about the causes of variation. No, they're saying about in meiosis. So what are you supposed to talk about here? You talk about the the random arrangement of chromosomes during metaphase. Yes, random arrangement of chromosomes during metaphase, and also talking about crossing over during prophase 1. Remember, when you talk about prophase, crossing over, you are specific to prophase 1. Prophase 1, not anything else, is prophase 1. You don't talk about something else. It's supposed to be prophase 1. That is crossing over during prophase 1, which leads to exchange of the genetic material. So please don't go to the paper without knowing the process of crossing over. Don't go to the paper without knowing anaphase. Don't go to the paper without knowing metaphase. Yes, metaphase 1, metaphase 2, anaphase 1, anaphase 2. Those we might likely to bring it. Division of a cytoplasm, what does it mean? Is called cytokinesis. This division of a cytoplasm is called cytokinesis. Lastly, on meiosis, what you can't go without knowing is know how to label. What is this? In most cases, people, they call this nuclear membrane. No, nuclear membrane cannot be seen. You see, the nuclear membrane can only be seen in telophase. Yes, yes, in telophase. And in prophase, prophase. So this is cell membrane. This is chromatid. This is centriole. The dot is centromere. This is spindle fiber. Ne? This is, what is this? This is a cytoplasm. So now I think you know. The last question whereby students get lost is draw the diagram of a cell at the end of meiosis, or draw the diagram of a cell after crossing over. Let me just show you. Let me just show you using this. Using this, I think I have the diagram here. Give the collective name, give the gender, name of observable reason, the main result of segregation, main result of segregation, describe the karyotype, Down syndrome will result differ. Okay, sure. That's not what I want. Yes, here. Look at that diagram. Look at that diagram, guys. If they ask you to draw the cell or this after this process, guys, look at where crossing over took place. In this case, the crossing over took place up, while here is took place here and here. So if they ask you to draw this cell, maybe they are saying chromosome X and chromosome Y, whereby you say structure of chromosome Y after maybe crossing over, then you have to draw it nicely. You have to draw it nicely. Yes, you have to draw it nicely. But where did the crossing over take place? Crossing over took place on this chromatid, but halfway up. So you mean that when I'm shading all this, remember this is the black thing, so I have to shade all this. I have to shade all this. Yes, I have to shade all this to be black. And then the crossing over did not start from here, it started from here. So this variation of shading will give you a mark. Writing a caption will give you a mark. And then don't forget to label. Even if they have not asked you to label, label. Even if you don't ask you to label, at least have 2. Because in most cases, when they ask you to label, at least 2 points. Yes, they say at least 2 points. You have labeled it at least 2 points. At least then you get those marks. Yeah, please don't forget to label. If they don't say, if they say draw a label diagram, or they'll say draw and then just leave it like that. Guys, make sure that you draw and label, because it's not losing you. It's not taking, they won't cross it because it's labeled. But you get a mark if you labeled, just in case, because you are not a memorandum to know which one is not going to be credited or is not going to be, is going to be credited. So you just write. Lastly, before I forget, if they ask you to describe, write everything you know in the head. At least for us, we look for what is correct for you. If they say name, that one you only name. You don't give alternatives. But if they say describe, just write everything you know. Write everything you know. Or sometimes I call it write all the rubbish. I'm not saying that write the rubbish, but write everything, whatever you have in the head. Don't leave it, because when you leave it, you come out of the paper and say, I thought about it. I thought about it. I thought about it. No, don't do the regrets, because this is your last time to change this subject, the mark. So just write everything. If the correct answer is there, we look for it and we'll give you a tick. Yes. Yeah. Do you have any question, guys? Comparative anatomy. Someone saying comparative anatomy. Okay. Thank you. Guys, if you have any question, you let me know, because I'm here to make sure that you get a distinction. You get a what? A distinction. Distinction. Yeah. Make sure that the distinction clicks, because we want it and we will get it. You will get it. Yes. God willing, you know me, when in my region we say, Inshallah, Inshallah, we will get a distinction. God willing, we will get a distinction. That's what it means. All right. Check this. Check this. It's simple. You guys, Meiosis, I'm winding up now. Ne? Make sure that you know how to label those diagrams. Make sure that you go when you know the process of crossing over. Make sure that you know the karyotype. Make sure that you know the prophase, anaphase, telophase. Make sure that you know when you go there, when they give you a diagram, it must be east to west. Those spindle fibers, they must be east to west, meaning that you have to turn the paper. Turn the paper. No one is going to ask you, why are you turning the paper? It's your paper. No one is going to ask you whether you turn it, whether you write upside down. Doesn't matter. Turn it so that you identify the things correctly. You understand? Yeah, these phases, they are confusing people to identify. Please don't make a mistake. It's the last time for you to identify what is it. Let's try to label this and then we see. How do you call C? Is the centromere. How do you call this? The point where crossing over takes place is chiasma. How do you call this? We call it chromatid. How do you call this? Because they are chromosomes and crossing over, we call it bivalent. Yes, we call it a bivalent or bivalent. Yes. Or you can call them, sometimes you can call them, they can correct you when you say homologous chromosomes. Yeah, but scientifically we call it a bivalent. Bivalent, ne? Or bivalent. Ne? Someone might ask me the spelling. Yes. So identify, okay, we have done that. Name two ways in which structure A and B is similar. Structure A and B. How are they similar? How are these two structures similar? They have the same size, ne? Same size and shape. Good. Number two, they have the same location of the chromosomes. Not same chromosome. They are not the same chromosome. If they're same chromosome, you'd have looked exactly the same as your father. Yes. So they have the same position, locus, ne? Same position of the genes. They have the same position of the genes. Describe the process at D. So they are looking at describe the process of crossing over 5 marks. The homologous chromosome come together. They cross over at a point called chiasma. They exchange the genetic material between two homologous chromatids, and then they separate, and then they form genetically different chromosomes. So basically, that's what you need to explain when it comes to that. I'm not going to use too much of the distinction material, but I know that you guys, you will make it. Lastly, before I go, where does meiosis occur in male? It occurs in the ovary to form ovum, testis to form spermatozoa, then in plants, ovary to form ovules, anthers to form what? Grain. That's it. Hey guys, I feel good when you guys are feeling good. Ne? Don't panic, ne? Because when you panic, you make me panic. Ne? Yes. So make sure that you don't panic. Ne? Yes. Make sure, I repeat, don't panic. You're gonna make it. Let's go to genetics. Genetics. What I'm supposed to talk about? Genetics. Genetics. So we have used 1 full hour. Remember, we said that we're gonna discuss this paper for 2 hours. Eh? Yes. So we have used 1 full hour. So let's go to the second hour, that is genetics, and 30 minutes, and then 30 minutes for evolution, and then we know that we have smashed. Guys, make sure that you get all the marks in DNA, all the marks in meiosis. Yes, guys, in genetics, we'll talk about genetics. Terminologies, you know, I'm not going to talk about the terminologies, but go when you know that a circle means a female, and then a box means a male. Is it clear? That's number 1. Number 2, when they give you a question, identify whether it's a sex-linked or a non-sex-linked. I repeat, identify whether it's a sex-linked or a non-sex-linked. How do I differentiate between the two? Sex-linked, they will mention it that is sex-linked disorder. Always read what is on top here. Is a sex-linked disorder. Is it clear? Make sure that you read it. If they say sex-linked disorder, definitely you have to use X and Y. Male, there will be X and Y. Female, there will be X and X. If it is not a sex-linked disorder, please don't use X and Y. However much they have talked about same and karabo, and you know that this is a male, this is a female. Please don't use X and Y if they have not mentioned that this is a sex-linked disorder. I think I'm clear there. Number 2. Let's do one example on sex-linked recessive allele. One example, sex-linked dominant allele. One example, non-sex-linked. The first one, identify whether it's a sex-linked or non-sex-linked. Okay, now we have identified here that is sex-linked. The second thing you need to know, guys, I repeat, and I'm putting down the pen and the mouse. I wish you could see me the way I'm begging. Please, let me come closer to the mic. Please identify whether is caused by a dominant allele or a recessive allele. I repeat, identify whether it's caused by a dominant allele or recessive allele. Because if it is caused by a recessive allele, the one who is homozygous recessive is the one who is going to be affected. If it is caused by a dominant allele, the one who is homozygous recessive is going to be normal. Let me show you using here. This diagram is saying. So it means that it's caused by a recessive allele. Ne? They said it here. Ne? So it means that the one who is shaded, they're saying that is affected with the disease. Therefore, this person is a male because the sex-linked is going to be that way. While this is a female, is going to be small d, small d. Is it clear? This one is a male, is going to be capital D, Y. Okay. Question. What about if it's caused by a dominant allele? Perfect. This is caused by a dominant allele. Yes. Okay, let me first finish here so that I don't confuse you. Now, let me go to the questions and see how many generations are shown here. It's 1, it's 2, it's 3. So there are 3 generations. How many kids listen carefully. How many kids are sons of 1 and 2? How many kids are sons of 1 and 2? 0. How many kids are 1 and 2? 2. Remember that this is not a kid of this. This is just came and married this daughter. And this one came and married that daughter. Therefore, there are how many? There are 2 kids which are coming from these 2. Is it clear? You see that this thing is not linking down. Is it clear? Yes. I'm done with that. Let's go to the third question. How many family members are in this phylogenetic tree? Family members. It's 1, it's 2, 3, 4, 5, 6, 7, 8, 9, 10. They are saying family members. Is it clear? They're saying family members. Don't say that this is not a family member unless you're telling me that your father is not among the family. It's a family member. But if they say how many kids for this, then I think now it's clear you know what you're supposed to do. Yes. That's the third thing. If they say that this cross with this, produce these kids, do a genetic cross, because we are in the second generation, so it's going to be P2F2. Is it clear? If they say this, it's going to be P1F1. I think I'm clear there. Yes. So I will talk about when I go to the crosses. Then we go to the next, what you're supposed to know. Yes. Can you please do a live later? Today I join. Can you do the live again later? Today I join. No, what you can do, if the live is, start from where you have started from, and then after that, join you will play the video, ne? Yes, before. Lindo Putle. Why are you removing? I didn't see what you are saying, sir. The sound at the back. Eh, someone saying there's a sound at the back. Ah, serious, there's a sound at the back. Okay, let's go to the next question. Okay, how do I find the unknown one? I'm gonna talk about it, my daughter. Um, yeah, guys, I hope the sound at the back is removed. Ne? Let me know. Let me know. Let me know if the echo sound is removed. I don't want those echo sounds. Ne? Yeah, it must be nice. All right. Let's go to the next question. How do I identify? Okay. Now someone is asking. I'm gonna go there so that we finish this. Ne? How do I identify one? To identify one, remember you have to first identify that this is caused by a recessive allele. Yes, if it's caused by a recessive allele, so what you need to do is okay, what you need to do is, okay, this one is XX, but this person is normal. Therefore, it's going to be capital D. I don't know whether it's going to be capital D, small d, or capital D, capital D. But they produced a kid who is sick. Therefore, this kid is sick with it. Don't just say sick. Sick with what? With the disease. Ne? So automatically, one came from the father, one came from the mother. Therefore, this mother cannot be like this because it gave rise to this. You see? Whenever they ask you to identify, make sure that you look at the kids. Ne? Yes, which kind of kids they have produced. Therefore, we have identified that because this kid is small d, therefore one came from the mother, so therefore the mother is like this. Ne? Now, what about the father? Remember, this is sex-linked disorder. Yes, if one came from the mother, then definitely another one came from the father. Therefore, the second X, the first X is like this, or the X is like that. Therefore, the father is affected with that disorder. Don't just say affected. Affected with the what? HIV, sickle cell, flu, corona is affected with what? With the condition they have talked about. Don't leave the answer hanging. Ne? Please, 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 please. Don't leave the answer hanging. Then the second one. Okay, someone saying, ish. Hey guys, just give me 2 minutes. Ne? Let me check why is it I hope the sound is gone. I don't know whether. Okay. I hope the sound is gone. Yeah, you let me know. Please slow down. I hope. It's okay now. All right, let's continue, guys. Let me know if the sound is fine. I don't know because I've tried to see that everything is right. I've tried to check. Let me check. Let me check here. I've tried to see that. Yes. Let's continue and see. Hey guys, I think that sound, I don't know, because we changed the system and the system I'm using is a different one from the main one. Yes. So I had to change the stations. All right, let me just continue. I hope at least you get something. Yes. Okay. I think now you have got it correctly. How do we identify this? Yes. Now let's go to the question, the next question. Then we are finding out. Identify the phenotype of individual 2. These ones I'm going to do the questions and then we see. The phenotype of individual 2. So we have said that this person is normal. Yes, is normal. A normal male or female is a normal female, or female without C. Yeah, that disorder. Then the phenotype of 1, we have identified that this person, because they have produced a kid who is sick, and the sick is like this, therefore one recessive allele came from here, another recessive allele came from here. And remember, this is a male. This is a male. Therefore, this individual is affected with seed. And then they are saying that identify the genotype of number 3. Genotype of number 3. Yes. Remember, this person is sick. Therefore, is going to give remember, is a female. It's going to give 1 chromosome from there. But because it's normal, therefore the mother gave her this. Therefore, it's going to be that way. Because they say the genotype. They say explain how individual 5 inherited disorder. Individual 5. Individual 5 is small d. Therefore, we're going to say that individual 5 got 1 recessive allele from the mother and then 1 recessive allele from the father. No. Individual 5 is a male. Is a male. Can't be from the father. Father is normal. The individual is male. So you're gonna say that the individual 5 is a male who is sick, and then therefore obtain a recessive allele from the mother. Where is these 2 marks? You have to tell us that individual 5 is affected with this disorder and is received 1 recessive allele from the mother, who is number 4. Therefore, I think that's how you can try to explain. But now let's go to yes, the problem comes in when it is caused by a dominant allele. Okay, let's look at it. When it's caused by a dominant allele, Puberty, whatever, is syndrome sex-linked. You see, they have talked about it, but it's caused by a dominant allele, isn't it? The diagram below shows this syndrome in the family. Okay, now we are stating how many generations are there? There are 3. 1, 2, 3. Sharp. How many females are in this phylogenetic tree or in this family? Females. 1, 2, 3, 4, 5. So 1, 2, 3, 4, 5. Is it clear? How many females were produced by Patty and Gabby? It's gonna be females. 1, 2. This one is not family member, not a kid of this. It's a family member, but it's not a kid of this. So it's only these ones who are the kids. I think I'm clear there. Then now we go to the next question, whereby they're asking you, they're asking you, they're saying that, they're saying that, okay, name the all right, let's find out. Remember now they're saying that this is affected, né? And then this is also affected, this is unaffected. But don't forget that this syndrome is caused by a dominant allele. Therefore, these people who are affected, they're going to be dominant. Capital D. Can it be capital D, or capital D? Sorry. X, capital D, X. Is it capital D? No, they used G. Sorry, guys. They used G. Please use the letter they have given you. Don't just use what you think is correct. Therefore, it's going to be X, capital G, X, capital G, X, small g, no, capital G, and X, small g. Yes. But now I need to find out, we have, what about this one? This one is normal. So if it's normal, therefore it's going to be small g. It's going to be small g. Therefore, this person can't be this, because this person, the kid, has a small g. Therefore, came from here. Therefore, I have to say that this individual is like this. Capital, small g. No. Capital G, small g. So that one is done. What about the female? The male. Remember, this is normal. If it's normal, therefore automatically is having small g. That's why this and this, they combine to produce a kid who is a female with the 2 recessive alleles. We are done with this. What about this? Is a female who is sick. Who is sick. Therefore, this female can't be homozygous dominant. It's going to be capital G and then small g. Why? Because one X came from here, then another G came from there. You understand? Yes. Now, how many, name the type of diagram is phylogenetic tree. They're saying how many female in this family? We talked about them. 1, 2, 3, 4, 5, 6. Yeah. Male in this generation have, how many male in this generation? Generation 1 have what is syndrome? Generation 1. Where is the generation? The first generation. Eh? The first generation. Remember, this is a generation 3. 1, 2, 3. Because they are looking for male in generation 1. So this 1, 2, 3. They're talking about generation 1. They're saying with the disease. 0. Answer is 0. Because it's caused by a dominant allele. And then they're saying that give the male in generation 1 have what is disease. Give the genotype of Gabi. Genotype of Gabi. We talked about it because they produce a kid who is not sick. Guys, please take note which one is dominant, caused by a dominant allele, which one is caused by a recessive allele. They will have to specify that is caused by a dominant allele. Is it clear? Males in okay, we're done with that. Aju and Pirusa have another 4th child. Give a phenotype of their sons. And Pirusa. Yes, so of their sons. Remember this is caused by a dominant allele. The moment it's caused by a dominant allele is easier to find. For the case of what? The son. And then remember this one, son. Ne? It's a Y, it's a Y. Therefore, because this one is normal, remember is recessive, is normal, therefore this one is going to be, both of them are going to be normal. Don't forget it's caused by a dominant allele. If it was a recessive allele, then they would have been affected. But the one who has the recessive allele is the one who is normal. And the one who has a dominant allele is the one who is affected with that disease. Is it clear? Yes, that is the only trick you need to know about that. The one is normal. Yes, the one who is normal is having a recessive allele for the dominant allele, if it's caused by a dominant allele. The one who is sick is the one who is having a dominant allele. You You understand? Yes. Explain your answer above. Why? Because they are normal. Because Anju, yeah, who is a female, is normal with the 2 recessive alleles. Therefore, it gives 1 recessive allele to the males. You understand? Yes. Therefore, they are normal. So basically, that's what we need to talk about. When you go to a cross which involves an autosome, a non-sex-linked, yes. Please, even if they talk about same and carao, please don't use X and Y. Yeah, if they say that this also is caused by allele R, so you just say R. It is homozygous. If it's heterozygous, is R, small r. You don't put them on X and Y. That is the major difference between the two. Yes. Okay, lastly, about blood group. Then after that, we talk about the cloning. How many blood groups, how many genes control blood group? How many genes control blood group? How many genes control blood group? Someone is asking me, sir, it started confusing you. Daily interesting videos. How am I confusing you? Let me know. Let me know. Huh? How am I confusing you? Oh, where are you confused? Yeah, in this video description. You're saying I'm confusing. Let me know where am I confusing you so that I can sort that out. Ne? Yeah, just tell me that maybe this question or this question. Yeah, so that I can sort it out. How many genes? Yeah, correct. Correct, guys. Correct. Thank you very much. Yes. How many genes control blood group? It's 1. It means that you guys watch my videos. Thank you very much. And I'm seeing live as Mbali, as a member. Yeah. Thank you very much, Mbali. Yes, for being a member. And I saw another member here coming. Oh, Lindy Wayne Love, thank you very much also for joining us. So you are on the team, ne? Yes. All right. Never be saying that X and X are. Oh, okay. Okay, let me explain to you what I meant there. Someone is saying that X and XR. I'm saying don't use X and. Okay, let me just look for a question here. Yes, here is a question. Ne? Mbali. I think it's Mbali. Ne? Yeah. Here they're saying that this syndrome in dog is caused by a dominant allele, capital H. You see? The pedigree diagram below shows the offspring produced by 2 dogs. You understand? They're not saying that this is a sex link. You understand? Yes. How many offsprings are affected by this disease? How many offsprings? You see now? Is caused by now this one is also caused by a dominant allele. Is caused, meaning that the one who is sick, check here, even here they're saying it, that dogs with the disease and dogs without the disease. So these ones which have the disease, it means that they are going to be capital H, or can be capital H, small h. Even this one is going to be capital H. You see, is a male, but I'm not using X and Y. Capital H, or capital H, small h. Whenever there is a capital H, because it's caused by a dominant allele, this individual is going to be sick. You understand? Yes. So, however much is a male, I'm not using X and Y. You understand? I don't use that. Why? Because they didn't say it is caused by a sex link. Is it clear? Yes. So in this case, but I can identify because they say that how many individuals are affected. Okay, that one is simple. But now this one is what is not affected, meaning that this individual is gonna be small h, because it's caused by a dominant allele. Whenever you see a capital H, it means that that individual is what? Is sick. If they produce a kid here who is having these two, then automatically one came from the father and one came from the mother. Therefore, they can't be this, they can't be this. So this is like this, so that one H comes here, and then this one is like this, so that another H comes there. You understand? So when they give you a question and they don't talk about sex-linked, please do not use, I repeat, do not use X and Y. Use the letters the way they have given them to you. Yes? You only use X and Y if the statement says that is caused by a recessive allele is sex-linked. You see? Is sex-linked. Even here they said it here is sex-linked. Even they gave you what to use. That is carried on X chromosome. Is it clear? Yes. So this situation is caused by a dominant allele, but it can be caused by a recessive allele. So I think now I'm clear when it comes to that difference between phylogenetic tree and the pedigree. Phylogenetic tree, guys, phylogenetic tree is explaining the evolutionary relationship among the members. You understand? Evolutionary relationship, like how did you evolve? But a pedigree is describing a character in a family. Is it clear? Is describing a character in a family. Yes. Okay, guys, let's go back to blood group. I think I've got it. You have got it. I'm no longer confusing you. How many genes control blood group, guys? Is one gene. Is one gene. How many genes control blood group? Is one gene. How many alleles control blood group? One gene, and that gene is blood group. Do you have any other gene? Tell me if there's any other gene. It's one gene, blood group. Yes. But how many alleles? There are 3 alleles. Yes, that is allele for A, allele for B, and allele for O. Guys, please don't make a mistake and you say allele for A like this. This is not A. This is a Roman numeral, which means 1. Allele for B is supposed to be like this. Allele for A. Don't put at here. Don't put them, make italics. Don't say allele for O like that. What is this? We will cross it. I is supposed to be like that. Is it clear? Yes. So allele for A, allele for B, and allele for O. So there are 3 alleles. Yes. I don't want to go to the distinction material to show everything. No. We go to the next part, of which the next part they're asking you how many genotypes. Genotypes. Oh, before I go, guys, back, there's something I forgot. Before I go back, I don't know what took me off. Why are males more affected by sex-linked disorders than females? Why? Or they can say, why are males more affected by hemophilia than females? Or they can ask, why are males affected by color blindness than females? So the answer you give here, if they say 4 marks, you say that males have 1 X. Yes. If a recessive allele, for example, is capital small h, appears on X, yes, the individual is affected with the disease, whatever they have talked about. While females, they have 2X. Yes. If a recessive allele appears, for example, on 1X, it can be masked by a dominant allele on another X. Therefore, the individual is normal. Males have 1X. Yes. If a recessive allele appears on the X, the individual is affected with that disorder they are talking about. While females they have 2X. If a recessive allele appears on 1X, it can be masked by a dominant allele on another X, and the individual is normal. Sometimes they can ask you a question that why males have higher chances of becoming hemophilic than females. So guys, it's the same thing. Please, you need to know about that. Ne? Yes, you need to know about that. We go to the next question. Because they have 1X. Yes, because because they have one X. This question they can come for 3 marks, ne? Yes. Is it possible for a person of blood group A marrying a person of blood group B and they produce a child of blood group O? Is it possible? Yes, it is possible. How do I know it's possible? You look at the genetics, ne? Okay, here the question is A. Before even I go there, let me just answer this question, at least one question, so that, yeah. A man who is blood group AB and then who is heterozygous, marry blood group B, and then have a children. How many alleles control blood group? They're now talking about the alleles. Yes? Gene 1 is 1. Allele, there are 3. Genotype, there are 6. Phenotype, there are 4. Why am I using 4? You have A, B, O, and AB. A, B, A, B. Do you understand what I'm saying? A, comma, B, comma, A, B, comma, and O. Those are the 4 phenotypes. But genotypes, there are 6 because A is homozygous or heterozygous. Let me show you. Let me show you the distinction material, because some people, in just talking, they might not take it, and they might take it long to understand my ideas. See, you see, here is what I'm saying from the distinction material. Blood group are as a result of multiple alleles. It's a multiple allele, yes, meaning that it has more alleles, more than 2. There are 3 or more alternative form of yes, this. There are 3. All right. So phenotypes, there are 4. 1, 2, 3, 4. You understand? And then the genotypes, there are 1, 2, 3, 4, 5, 6. That's what I say. Because this is homozygous and heterozygous, homozygous and heterozygous for dominant, and then homozygous recessive. Yes. Then they're saying that 3 different alleles, which are this, and then 4 blood groups or types, which we call the phenotypes. No, the genotype, the phenotypes. Yes. So it's A, B, O, A, B, and then O. That's what I was saying. A, comma, B, comma, A, B, comma, and O. So that you understand. Yes. So now we go back to our question. Describe the type of dominance that occur in the inheritance of blood group of woman B. Describe the type of dominance that occurs in the inheritance of blood group B. Now they're saying inheritance of blood group B. Check. They're saying that a man with blood group AB and a woman who is heterozygous for blood group, yes, heterozygous for blood group B, meaning that this lady is, it looks like this. See? Yeah, meaning that obtained one dominant allele from the father or mother and another recessive allele from the father or the mother. So it can come either from the father or mother for A, and this either father or mother, because they are not specific in that regard. Yes. Use the genetic cross to show the possible genotypes of the phenotype of their children. So you guys here, this principle works to all of them. Start with P1, F1. Please, F1. Don't just say P1 and then you leave, you finish after you say F1. You will forget this F1 and then you lose a mark. So you say P1, F1, at least we shall come here and give you a T. Is it clear? Then you say P1 phenotype, because P means parent phenotype. Ne? Yes, you tell us. The phenotype is always obtained from the question. Check here. You use, because these 3 marks, it is 6 marks. 3 marks are mahala. You understand? Use the genetic cross to show the possible genotype of phenotype of what? But what did they say? They said a man who is blood group. So you say a man of blood group AB times a woman of blood group B. You don't need to tell us heterozygous, because when you look at someone, you can't say that he's heterozygous. We just say heterozygous, homozygous, so that you know what you're going to cross. So you're saying P1, F1, we give you a tick. Now you tell us phenotype, you tell us a man of blood group AB times a lady of blood group B. That's all. Then we give you another tick. Why? Because you have identified it, but you always get it from the question. So that's the second mark for Mahala. Then you talk about genotype. If you don't know the genotype, if the space, talk about meiosis. If you don't know the meiosis, if the space, talk about gametes. If you don't know the gametes, leave the space. Then you talk about fertilization. Don't know this fertilization, leave the space. But you shall come here and give you a mark there. So you have got 1 mark, 2 marks, 3 marks. Meiosis and fertilization, they must go together. If you know now, that's where you're going to obtain the other 3 marks. To know the genotype, because most of the cases, you people, you get lost on the genotype. When you get the genotype wrong, the remaining thing here becomes what? Becomes wrong. Someone is asking me, please, I answered that question. You have to rewind the video. Yes, I answered that question. Eh? Yes, for case of the DNA changing the triplet. Yes, I answered that question very well. Please, you have to watch it maybe when I'm done with this session. All right, guys, let's go to the next question. That is dihybrid cross. Then I show you what you do or what you focus on dihybrid cross. Dihybrid cross, we are not going to ask you to cross. No. What we might ask you about is identify the genotypes of the parent, the phenotypes, and the gametes. Ne? Yes. So guys, you need to know first identify which one is which. Ne? But now if they ask you what is the genotype of the parent here, for example, ne? Don't repeat the letters before you finish. For example, the genotype of this can be capital P, even capital L. Then another one is capital small p, small L. So the one which are the same, you combine them. Capital P, capital L, small l. So this becomes the genotype of the parent. I'm just trying to show you how do you identify. And if they ask you to identify the gametes from here, then it still is fine. You will say that P goes with L. P, capital L. So this one is done. Now P again with the small l, capital P, small l. So it's done. This with this now, small p, capital L. Then this with this, small p, small l. Sometimes people, what they do is not a cross. It's not a cross, but what they do this. They do this, capital P, small p, capital P, small p, capital L, small l. They just cross with this, capital P, capital L. They cross this now again with this, capital P, small l. And now they cross this with this, small p, capital L. And then now this with that becomes small p, small l. See, they form the same gametes. You understand? Yes. So at least you have done. Now they can ask you, they have given you the gametes. They want you to write the genotype. Yeah, that's where the story comes from. So you start with you start with one letter, capital P. Do I have any other capital letter? Yes. So you first leave it. Ne? Do I have a small letter of the same type? Small p? Yes. If I have a small p, I write the small p there. So they are supposed to be 2 letters. But if I don't have a small p, then definitely I will have to write a capital letter. Is it clear? I will have to write a capital letter here. But because I have a small p, therefore I will have to write the small p. Then I go to L, capital L. Do I have another capital L? Yes. Do I have a small l? Yes, I have a small l. Therefore, it's going to go with small l. Therefore, this becomes the genotype of the parent. Check. It's the same thing. Capital P, small p, capital L, small l. Then when you come here, it's capital P, small p, capital large L or capital L, small l. So that's how you can identify that. Difference between transcription and DNA replication, I talked about it, guys. I talked about it. Just wind up. I know that you will find everything you have asked me. Ne? You are trying to ask me. Yes. Lindo, yes, I talked about it. Difference between transcription and DNA replication. Yes. And even the similarities also talked about then that both the DNA double helix unwind. They talk about both there is unzipping. Yes, when you talk about differences, you can talk about DNA is formed, RNA is formed, two templates are formed, one template is formed. So you guys, we talked about it, né? Yes. All right, let's continue to the next one. How many genes control? Once you see the dihybrid, then there are two what? How many genes? There are two genes, né? Dihybrid is controlled by two genes, né? Yes. How many alleles? Yeah, in most cases, there are 4 alleles. Yes, you'll find out the capital letter and the small letter. So depending on whether you see there here, it's 1, 2, 3, 4. But there are two genes. Genes for the number of seeds per pod and then the shape of the what? The leaf. So guys, the hybrid is gonna be is simple, né? Simple. Just make sure that you do it, né? Yes. So it's the same thing here. No one is gonna ask you to cross. Before I leave this point, I wanna ask you a question. Is it conclusive to use blood group to identify the paternity, the paternity of the individual? Is it conclusive? I want yes or no. Is it conclusive? Then once I see the answer, then I will give the reason. If the answer is correct, I will give a reason why. Three factors of, okay, the experiment, that's one you must know, guys. Lastly, let's go to, before I go to, please read about the stem cells, read about the cloning. Yes, cloning, the process of cloning. I designated a full video on cloning. Ne? Stem cells, what are stem cells? The cells which have ability to modify and form other body structures. Yeah, basically you just need to read this and the examples of these stem cells. Yes, let me just show you here from Excel and Life Science Distinction Material. Yeah, see sources of bone marrow and umbilical cord, adipose tissues, amniotic fluid, uses, yeah, to generate new or replace the worn out cell or damaged cells, or to generate new cells, ne? Be used for research purposes. Yeah, so cloning, please know how to use this cloning, but most especially, let me just talk about, yeah, it can come like this or it can come as a doll. What? Do you know that this is the name of a person? Is a singer. Dolly is a singer from, I think it's from America. Do you know that? That is a singer from America. You just listen to people's songs, but you don't know that they're asking you questions and you don't know. I hope he's a singer, eh? I'm not a fan of. Okay. Is it conclusive? Okay, thank you very much, guys. Everyone is saying, sir, can you please touch on stem cells? Okay, sure. Someone is yes, I'm saying that. No. Yes. It's no. It's not conclusive because many people share the same type of bloody group. Therefore, it's not conclusive. It's not conclusive. Yes. It's not conclusive. Someone say, yes, I'm going to talk about them. Let me just talk about stem cells. Then we shall go to, not stem cells. I'm done with stem cells. We talk about the cloning. Guys, this is what I want to clone is this. I want to clone. Let me just do like this so that it gives me the full version. I want to clone this. Therefore, I have these two. I get ship A. If I want to clone this, I have to keep its nucleus. Is it clear? Why? Because the nucleus contains the genetic material. So I keep the nucleus, I throw away the shell. Then this one, I remove the nucleus because I don't want its genetic material, but I use the gamete, the ovum. But here I just get any cell from anywhere else apart from a gamete. So I throw away the nucleus because I don't want it, but now I keep this. But why do I need to have, why do I have to be with a casing of the cell? Why should I be with the casing? Yes, it's like you drinking water in a bottle of alcohol. What are we going to say? We shall say that you're drinking alcohol, Baba. Yes, that's what you're going to say, that you're drinking alcohol. Yes, Why? Because you are putting water in a bottle of alcohol. So when I put this nucleus into a cell, yes, this cell, it will look like a gamete. Remember, this is a diploid cell from anything else. So if I put this nucleus to this, then this one is going to look like a zygote, because this is a casing of an ovum, and I've put a nucleus which is 2n. You understand? Therefore, it's going to look like 2n. So if they ask you how many chromosomes are here, these are, if they say the sheep has 24, therefore it's going to be 24, or it's 2n. Yes, you understand? Yes. So now, we are saying that which method do we use? We also ask you about the method. Is mild electric shock. Then you have here the foster mother here, and then we bring this, we put it in here, and then after putting it here, then this one starts to develop, develop that embryo, and then it forms the baby. This baby is exactly the same as this. You can ask you a question: why do we remove the nucleus? Why do we keep the nucleus? Because we wanted the genetic information from sheep 1. Why did we throw the nucleus away? Because we wanted the casing of the ovum. We are not interested in the genetics here. Yes. So which method we use? Mild electric shock. And then which sheep will this, which of these two sheep will this one resemble? Sheep 1. Give a reason for your answer, because we use its the nucleus, we use its nucleus or the genetic material to produce the sheep. Yes. So when they ask you such questions, they're not that difficult. You'll be able to answer and get it right. Paternity test, blood group. So it's the same thing we are saying. This is the question I was asking. Is it conclusive? It's not conclusive, because many have the same type of blood group. So it's not conclusive to confirm or to determine that this is, yes, the parent of the child. Guys, now we are going to, first of all, this is the topic I'm talking about, reproduction in vertebrates. What you need to look at here is, number 1, you need to know what you call external fertilization, internal fertilization. Once you differentiate between those two, then the whole topic, you are done with it. So internal fertilization, when you talk about internal fertilization, here we are talking about the sperms must fertilize the ovum inside the female's reproductive system. You understand? Inside the female's reproductive system. And then when you talk about the external fertilization, the sperm will fertilize the ovum outside the female's reproductive system. Yes, we say female's body. Sometimes we ask you where exactly in the female's body, inside the female's reproductive system, or where exactly it takes place inside the female's reproductive system. So you need to be specific where exactly is taking place. Female's body is also correct, but the most common one is inside the female's reproductive system. So that's number 1. The moment you know that, then you'll be able to understand another thing which we call what are some of the advantages of internal fertilization. Why should we have internal fertilization over external fertilization? Here it is stipulated well that internal fertilization increases the chances of the gamete, yes, these gametes or the gametes to have a successful fertilization. When the sperms are put into the vagina, they flow. When they flow, yes, they move towards the ovum. This ovum, the chances of meeting the ovum are higher than the chances of not being into the reproductive system. Imagine you just pour it outside. Then it means that the chances of finding the ovum will be minimal, because some sperms will go this side, some sperms will go the other side, others that side, others that side. So you end up losing some sperms. But if it is inside the female's body, female's reproductive system, the chances of getting fertilized, the ovum, is high. That's why we say that it increases the chances of meeting or mating, greater chance of successful fertilization. Increased chance gametes meet and greater chance of successful fertilization. So basically, that is the advantage of internal fertilization. Question: once you know that fertilization, then you will know these 4 terminologies. Once you know those 5 terminologies, the 8 marks are covered. There is nothing they're going to ask you too much about this. Let me show you what I'm saying. If you don't understand, let me know. This is what I'm saying. You have oviparous. When you talk about oviparous, what does it mean? When you say oviparous, you see it's going to be talking about fertilization. Yes, yes, they have the eggs and so they are laid outside. But here, yes, you will find out that the fertilization for oviparous is outside. There is no, as long as the fertilization is outside, don't think about anything else. Don't talk about viviparous. Don't talk about oviparous. No, oviparous. You don't talk about that. You only talk about when, as long as external fertilization, that's it. Outside, oviparous. Is it clear? Yes. I'm not going to explain too much, but I'm just telling you what exactly. So when they ask you what is oviparous, or in terms of terminologies, yeah, this is whereby the development of the embryo is outside the female's body. Yes, it's outside the female's body. Basically, it's outside the female's reproductive system. That's it. So you see these chicken and stuff. Obviously, once you see the egg, automatically that anything which lay eggs, that's oviparous. Anything which lay eggs. If they ask you an example, don't think too much. Anything which lay eggs, that's it. You can think about it. Anything which lay eggs. Yes. So does human lay eggs? No. The egg stays inside. So either the human is viviparous or ovoviviparous. You understand? Yes. So let's go to the next part. Vivi means inside. Vivi means what? Inside. So that one is clear. So the development is inside the female's body, and the key point here is the embryo obtains nutrients from the mother through placenta. Placenta. If you don't see the word placenta, then automatically it's not right. It must not have the word hatching. You understand? So that's it. So that one is clear. Then it means that here you give birth. You don't hatch. You don't lay eggs. You give birth. That is the key point. That's why you are seeing it there. You give birth. For example, humans. Let's tell us with the things there. Think about anything which give birth. Not lay eggs. Not lay eggs. Number one. Not what? Not hatching. Yes. Because when you talk about hatching, you will be talking about the chicken. What? Automatically the egg is there. The egg is there. Yes. Then ovo means ovum, vivi means inside. It means that the ovum develops inside the female's body. The embryo develops inside the female's body, but the nutrients are obtained from the fetus through placenta. Not through placenta. From the embryo obtains nutrients from the egg yolk. That's why here you see that we obtain nutrients from the egg yolk. That word egg yolk is very, 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 very important. Yes, egg yolk is very important. So that one is done. So you see now the 8 marks are gone. You understand? Yes, because we want to rush to the other side. So the last part of it is what are advantages of laying. What are advantages of having the baby developed inside and the baby developed outside, meaning the embryo developed inside and the embryo developed outside. So advantage of viviparous over oviparous is this developing embryo obtains what you call protection. It is being protected. They are not exposed to predators. Imagine you are walking on the street and then you how do you call it? You slap a pregnant lady. What is gonna do with on the womb? Automatically the lady is gonna have to fine you or do something. You understand? So why are you doing that? Why is the lady doing that? To protect the developing embryo. You understand? The embryo is innocent. You see? Yeah, so that's the protection we are talking about. So basically, that's it on this. The last terminology before we talk about the egg is precursor. Pre means prepared, born prepared. You have the eyes open, you can move, you can walk, you have down feathers. Yes, everything prepared. You are prepared to come to this world. That's the meaning. Born prepared. That's why you're saying that young are mature and able to move directly after birth. That's it. And then the eyes are open. These are the characteristics. So they will bring diagram. Oh, diagrams can be there, and then they ask you to identify which one is precursor. Give the reason for your answer. Which one artificial? Give the reason for your answer. You understand? Yes. And then artificial. This one means that all depends on the parent. Yes, all artificial. All depends on the parent. Meaning that you are helpless. Eyes are closed. You don't have down feathers. You will see that it's helpless. You see if they bring a diagram like that and ask you what is it? Is it precursor, artificial? You see everything. This baby is helpless. For example, you. Let's drink for that. An example, what? Yes, yes, we can talk about birds here, but even you. How many years do you take for you to be able to sustain yourself? So basically, let me do a recap about this. The importance of this is still increases the survival. Lastly, this one terminology from these parts, you might find it there if you are lucky. If you are not lucky, you won't find it, but I know that you are lucky. Amnion, chorion, allantois, yolk sac. Guys, I know yolk sac is a sac, but sometimes they ask you which part sir, is this prediction for June? Which part is they can ask you that which part provide nutrients to the developing baby. You're gonna say yolk. In actual sense is a yolk, but in exam we don't want yolk. You have to say yolk sac because we're talking about those membranes. The four membranes, which is amnion, the outermost membrane. You can ask you that question. Outermost membrane surrounding the embryo is called the chorion. Is it clear? The outermost membrane. The amnion is that fluid when you are dancing. Ne? Yes, in the baby. Ne? When the baby is dancing, you call it what? Amnion. Yes. Allantois, this one is is in these eggs. We don't have it in the babies, in the human babies or embryos. This one is very important for storage of wastes. You understand? Yes. And then yolk sac, nutrients. Nutrients. Basically, it's nutrients. So, guys, make sure that you know how to differentiate. And all these membranes are called what? Are called extra-embryonic membranes. Yes. So these are membranes which basically make up the what? The embryo. Yeah. So lastly, on this, before I wind up here, you need to know the following. I repeat, our 8 marks. The first one, internal external fertilization. That is, you have to know the example. Number 2, the chances of advantages of internal fertilization. Guys, are you listening carefully? Advantage of internal fertilization. Advantage of internal fertilization. What do you mean by oviparous? We say that is laying eggs and the embryo develops outside the female's body. Viviparous, we say that your development is inside the female's body, and it means that here the baby obtain nutrients through placenta, and birth is linked there, meaning that you give birth. When you see give birth, even don't think about it, is viviparous. Then we say oviparous. Here the development is inside, but the baby obtain nutrients from the egg yolk, basically the yolk sac, as we talked about. Yes. And then what are advantages of developing inside over developing outside? So that is also another point which you need to know. We say that basically is for protection. Yes. Then I say that one terminology from here might come. Listen carefully, might come. Say, okay, let me, sometimes when you laugh at a student and then you know something and that, make sure you focus on it. Then after that, sir saying this, yeah. I don't want you to be like that. Ne? Yes. Hi, sir, I'm from Soweto. Yes, I'm also from Soweto. It's nice to see you guys, ne? It's nice to see you guys. Or to hear from you. If you have guys any challenge, make sure that you are able to obtain these distinctions. Ne? Let me know if you have any challenge. So guys, I said that make sure you know these terminologies I talked about: amnion, chorion, alontois, yolk sac. And I told you that this one we ask it too much in terms of what is the outermost membrane which is surrounding the embryo. We say that it is the chorion. We say that the membrane which nourishes the baby, the developing embryo, we say is yolk sac. Don't say yolk because we want yolk sac. Yes. So we say that basically this one is alontois. We say that alontois is very good in storing the waste product. When you break the egg, what you find there smelling, that's it. That's alontois. Precocial, born pre, which means prepared. Eyes open, everything is there. Then atricial, we say that. This one all it's born helpless, meaning that it depends on the parent. So you need to know also that. Lastly, parental care, we say that the parental care, you don't need to read a lot. What I'm saying is, yeah, that's exact. If you want more than 50, yes, you understand. Parental care, what is it for? Is basically to teach the animal and basically for protection. That's it. This it helps to teach the habits of the animal. So the most important thing is to increases the chances of the offspring to survive. That's protection. Yes. Also teaching you the habits of these organisms or these animals. You understand? So guys, 8 marks gone. Yes. I'm writing on the 17th of June. Yeah. You get a distinction. Human reproduction. I want to ask you a question, guys. Should I go through human reproduction? Are you trying to mean that to tell me that you don't know human reproduction? Don't lie to me. Stay virgin. If you lost it, start now. Except when you are married. All right. All right, guys. Let's start with human reproduction. We say that. Check here. I don't see you giving me answers. I'm only seeing some few people. Glycogen. Good. Thank you very much for those people who have tried to give me the answers. Yeah. Let's do it together. Yeah. The reproductive strategy where young receive nutrients through placenta. You see, the word placenta. Do you see what I told you? Yes. The word placenta. The word what? Yes. They're saying that they reproduce placenta, which shows that you feed through placenta. Then it means that that is the VV paras. Duct that transports semen, urine to outside part. This one we're gonna look at it, but it's called vas deferens. Someone said epididymis. Epididymis, we're gonna look at it. What is the difference? But it's called vas deferens. The part of the nervous system that consists of the cranial and the spinal. These are other parts, not central, but the other parts. So it is peripheral nervous system. The form in which excess glucose is stored in the liver. Glucose is stored is someone saying sperm duct. We use vas deferens. Don't use sperm duct. Thank you very much. These are some of the reasons why I gave you so that I see what you are writing. Someone said, okay, I'll reach there. Sperm duct, please write vas deferens. Saying that the part, someone saying ovipary. Okay, yes. They're saying that the structure that serves as excess glucose is glycogen. Yes, that's how glucose is stored in a what? That's how glucose is stored in a body. Is glycogen. Yes, someone said it. Clement. Thank you very much. And then they're saying that the pigment, the structure that serves as a microfilter during pregnancy. How do you call that? That is a placenta. Yes. Someone saying it. Kiarebo Hire. Yes. Kiarebo Hire. Thank you very much. Yeah, it is a placenta. Then they're saying that the pigment, the pigmented layer of the eye that absorbs excess light. That is the choroid. If you don't know all these things, I'll show you exactly how to get it. They're saying that the part of the neuron that plays a role in speeding up, in the speed at which the nerve impulse is transmitted. Yeah, what determines the speed is the myelin sheath. Someone said it, but you are sheath is a sheath, not a sheet. Yes, I'm seeing something there is sheet, something like that, but it's supposed to have an H at the end. So I think you meant to do that, but when you're marking, guys, we have to look at what you have written, and in most cases, if it has a meaning somewhere else, you lose it. Yes. So the part of the male reproductive system where in which the part of the male reproductive system which temporarily stores sperms until they mature. That is where are the sperms stored? Someone is saying choroid. Yes. Yeah, sperms are stored in epididymis. I think 2 point. Yes, epididymis. Epididymis. Yes, that's the correct answer. Thank you very much, D. Daily D. I think yeah is a right one. Yeah, all right. I think let's go back to reproduction. So I've tested you. I'm gonna test you again. Yes, thank you very much. All right, human reproduction. What are you supposed to focus on? The first thing you need to focus on so that we can score our 41 marks, our 41 marks, is label the parts. Know how to label. That's obvious. I've found out that many girls know this structure more than the boys. I don't know why. You girls, you have to tell us why. I don't know why, but you have to know. Tell us. You see, this is it. Where is it? Yes, vas deferens. We call it vas deferens. Please don't call it. Don't call it. Please do not call it sperm duct. Yes, because it's not the only duct of sperm in this male reproductive structure. Yes. So please call it vas deferens. This is where sperms are stored, which we say that it is the epididymis. It's called epididymis. Then this is a testis. This is a scrotum. This is basically for protection and controlling the body the temperature of the testis, for skin sensitivity, urethra, transportation of the semen. Yes. Please, can you go back to 1.2.1.2? What's happening with it? 1. The duct that transports semen and urine out. Oh, the duct that transports semen and urine outside, yes, is called urethra. Yes, this is the part I'm talking about, urethra. Yes. Thank you very much. Thank you very much. Sozo. Yes, Sozo. Yes. This one is saying peripheral nervous. Yes, correct. Sozo, thank you very much. Yes, it is called urethra. I think I skipped it. That's where I was also talking about now. All right, guys, make sure that you know this view. We like it because you know that you fail it. You know which parts do you fail? Yes, you fail the cowpass gland, the seminal vesicles, and the prostate gland. Those three glands are called accessory glands. Are called what? Accessory glands. These are glands. Let me show you how to identify them. If they bring them, let me zoom it out so that you are able to see. Look at this. If you start from the outside part, the first gland you find is a cowpass gland. Then you go, you'll find the second gland, you see, which you call the prostate gland. Basically, is the biggest gland in there. And then the last one, before you go inside, is called, it actually sometimes it looks like this. Looks like that. We call it the seminal vesicle. This one, the seminal vesicle, because the sperms are coming like this, it gives the sperms the nutrients so that they're able to swim, the energy. Then the prostate gland, this one, it provides alkaline fluid. And then prostate gland, cowpass gland, the one which is outside, it gives the sperm a lubricant. It gives a what? Gives a sperm, it makes it to be lubricant, so that when it's moving out, it's very fast. I don't know, yeah, but that's how it's supposed to be. Yes. So, and then we say that the prostate gland gives the alkaline fluid, which neutralizes the acidic nature of the vagina. They're going to ask you a question: which gland produces a fluid which neutralizes the acidic nature of the vagina? Or what is the function of the fluid produced by the prostate gland? Yes. So, in this structure, you guys fail this male reproductive structure more than the female. And you boys, you know the female more than the girls, while the males, they know the female more than the what? Yes. Anyway, that is it. So guys, the functions, I'm not going to talk about that much. Just know the structure and the function. The moment you know the structure, when they give you this diagram, label everything. You understand? Yes. The only part which confuses you is seminal vesicle, cowpass gland, and prostate gland. The rest, I think, is clear. Yes. So, here they're going to ask you a question. Why are the scrotum? Why are the testes? We call them testes. Some of you call them other names. If you call them other names, it's up to you. But you call them what? Testes. What is the difference in testes and testes? You have this. Yes. It's the same as that, but this is many testes. Yes. All right. Why are the testes hang outside the female's the male's body? Why do they hang outside? They hang outside to provide favorable temperature for sperm production. That temperature is 2 degrees less than the body temperature, meaning that around 35 degrees. Sperms are produced at a temperature lower than at a temperature 2 degrees lower than the body temperature. That's why you have to hang outside. When it's cold, yeah, outside is too cold. Now they have to go a little bit up. I wanted to say something about if you get married and you find a man in cold and he's big, hey, run away. Anyway, in cold is supposed to contract. It's supposed to be small. While, sir, can I get genetics? How can you ask genetics in paper one? Yes. While in summer, when it's supposed to be now big. Yes. So guys, that's it. Why do they hang outside? To provide favorable temperature for sperm production, which is 2 degrees less than the body temperature. Don't say that because you're right to say that 2 degrees less than. Less than what? 2 degrees less. Less than what? 2 degrees less than the body temperature. Guys, by the way, don't forget to subscribe, like, and also share. Like the video. Like the video. You lose anything when you like? Okay, when you like the video, yeah, you make me happy. All right. Someone, Christina is saying is a female. Is saying that yes, I know the male structure. What? Yes. So if they bring the male, make sure that you pass it. Ne? Yeah, make sure that you pass it. I don't know. You're a moving textbook, but you don't know yourself. Yeah. All right. Functions of testosterone. Now the second function, the second thing they here to ask you is, what are the functions of testosterone? For secondary sexual characteristics, number 2, stimulates the maturation of sperms. That's it. Secondary sexual characteristics stimulates the maturation of sperms. The 3, seminal vesicle, prostate gland, Cowper's gland, we said we call them accessory. We talked about it. So that one is clear. So now I'm repeating before I go to the female. What do you need to know about the male reproductive system? Know the parts, how to label it. This view is very common. I'm not saying that it's the only one which is going to come, but make sure that you know it. You understand? Yes. Number 2, know the function. Please don't go without, because these ones, they confuse you. The 3 accessory glands. However much, yes, you can bring others, but those 3 accessory glands, they confuse you more. Why are the testes hung outside? They hang outside to provide favorable temperature for sperm production. Yes, which is 2 degrees less than the body temperature. That's it. What's the function of testosterone? The function of testosterone is to for secondary sexual characteristics, development of the secondary sexual characteristics, the characteristics which show that you are sexually mature. You are not mature in the country, but you are sexually mature. When you tamper with sex, if you're a lady, you're going to become pregnant. If you're a man, you're going to impregnate. That's it. That's the meaning. Yes. Then number 2, maturation of what? Maturation of sperms. Cool. Here, structure of the seminiferous tubes, you need to know only two things are needed there. The cells of Leydig and the satellite cells. That's it. Satellite cells are very important. What is it for? And cells of Leydig. We bring this in terminologies. Cells of Leydig, they produce testosterone. While satellite cells, they are very important for nourishing the developing sperms. Can you write the spelling down? Cells of Leydig. I don't know whether you say cells of Leydig. I don't know. Ne? Cells of Leydig. You understand? Yes. And then satellite cells. For what? Cells of Leydig are found here, and then the satellite cells are found here to nourish the what? The baby. Guys, you cannot go to the paper without knowing how to draw the structure of a sperm. How do I draw the structure of a sperm? And what do I need to know? And then once you know that, now the male reproductive structure is done. Even if they bring it like this, you're going to score high. Structure of a sperm. And I'm going to show you the question we ask you there. Let me show you. Structure of a sperm. Remember, sperm is like you. Yeah? I always tell you that you're a moving sperm. Don't quote me wrong that I said that you're a moving sperm. Don't quote me what? Don't quote me wrong. Yes. Oh. I'm saying that you're a moving sperm. You have a head. The sperm also has a head. I'm just drawing a simple structure. You have a head, the sperm also has a head. You have this neck, the sperm also has a neck. Small neck. You have this chest, the sperm also has middle piece. You understand? You have the legs, the sperm also has a tail. I'm just drawing it because the space is not enough. So you see that you're a moving sperm. On the top of the head, ne? You see now you are able to remember, ne? When you see the head, ne? You have hair. The sperm also has something here, which looks like the hair. Then inside the head, you have the brain, which control your activities. The sperm also has the nucleus, which control the activities. You see? In the chest, that's where the energy comes from. That's where the energy comes from. The sperm also has the energy in there. the structures called mitochondria. Use the legs to walk. The sperm also has the tail, which is used to. So when I say that you're moving sperm, am I wrong? I'm just asking. Yes. And don't be offended because you didn't come from a sperm. So this is a middle piece. You know how to do. It's a middle piece. This is a head. A, B, C. This is acrosome. D. This is the nucleus. E. This is inside there. You have the obvious, the cytoplasm. This is mitochondria. F. Yes. Now let's label together. A, head. B, middle piece. C, mitochondria. Ne? D, tail. E is nucleus. D is what? Acrosome. You understand? Now, from there, once you know how to label it, then now you need to know the function of each part. You need to know the function of what? Of each part. Yes. Let's look at the function of each part. The tail is to provide the propulsion force, forward movement. You understand? That's cool, I think. Yes. That's cool. Isn't it cool? Then the middle piece, yes, contains mitochondria, which provide energy still for movement. Isn't it? Yes. And then now we go to the head. The head has the nucleus, which contains what? Genetic information for inheritance, and also nucleus controls the activities of the cell. Then also has the acrosome, which this acrosome is responsible for what? Is responsible for it contains enzymes which digest the membrane of the ovum during fertilization. Listen carefully. Acrosome does not digest the membrane of the ovum. These are the mistakes my students outside there do. That acrosome contains enzymes. No, acrosome digests the membrane of the ovum during fertilization. It's wrong. Acrosome contains enzyme. The enzymes are the one which digest the membrane of the ovum during fertilization. Acrosome is the mouth. You can't tell me that the mouth is the one which break down the food you put in. No, the mouth has teeth. The teeth are the one which break down. That's why if you don't have teeth, you eat like, isn't it? Yes. So the acrosome is the mouth. The teeth is the enzyme which is found in the acrosome. I think I'm clear there. So when they ask you, acrosome contains enzyme which digest the membrane of the ovum in fertilization. So what will happen if there is no acrosome? It means that the sperm will not fertilize. What will happen if the flagellum or the tail is short? It means that it won't move faster. What will happen if you see? So we can bring an abnormal sperm and a normal sperm, and then we ask you some questions. Do you understand? Do you understand, guys? Yes. So that is the moving sperm, and that is the I don't know who are you. Okay, someone is asking me. Oh, all right. All right. Let's continue, guys. Ne? Yeah, let's continue to. So the male reproductive system is done. There is no way they're gonna ask you a question and that question is not covered there. Guys, this is a topic I repeat. You're supposed to score marks. You lose it, you have lost the paper. And the things I'm talking about now are the things you remember when they come. Yes. All right. Female reproductive structure is simple. Sometimes I call it, when you're like this, it means that, ne? It's simple, ne? The first one is the vagina, sometimes it's called the birth canal. You can ask you the second name is birth canal. Vulva is the outside part of the vagina. Yes, when you squat, what you see outside is not a vagina, it's a vulva. You understand? Yeah, the vagina is where the I don't know how you call it. Me, I call it zobza. That's where it goes. You understand? That length, that's what you call it. Because when they point it, you end up naming the vulva the vagina. It's wrong. Then the cervix is where the vagina separates from the uterus. You understand? So if you look at this, all that is the uterus. However, the uterus is divided into 3 parts. The first one is the uterine wall. The second one is the uterine cavity. The third one is the endometrium. So the endometrium, the uterine cavity, the space where the baby grows from is called the uterine cavity. When you call it uterus, still is fine. As long as they just labeled one. Uterine wall, this one, if you call it uterus, still is fine. They will give you a tick. You understand? Yes. So guys, make sure that at least if they label all of them, then that's when the challenge comes from. You understand? Yes. So this is fallopian tube. Fallopian tube, that's the site for fertilization. Ovary produces 2 hormones, estrogen and progesterone. Uterus is a site, that's where the developing embryo is situated, space where the embryo is situated, and implantation takes place on the other part, on other side of the, on the other side, on the other part of the uterus, which is the endometrium. So it can, even this one is the uterus. This one is the uterus, this one is the uterus. They can ask you a head. So wait, this is not a head, eh? Because this is the head. Yeah, it has different sides, yes. So 1, 2, 3, all of them are uterus. Cervix basically is protection and also very important, very important during the time of birth. And that's where the pain comes from. Yeah, don't want to talk about it, né? Yeah, because we just want to focus for 100% pass rate in life science. Né? Yes. Functions of parts. Guys need to know the function. I've talked about it. Né? Yes. Here, fallopian tube, site for fertilization. That's it. Ovary, I talked about, produces 2 hormones, progesterone, estrogen. You don't need to waste the time, guys. Né? Uterus, we said it carries the developing baby, the embryo, and the fetus. It's the one which carries, yes. The vagina receives the penis. It's obvious. How can you fail the function of a vagina? Unless you are not giving birth. Where did you pass? Don't tell me that is C-section. All right. I'm talking about then where did you pass when you are going there, where you came, where you passed when you are going out through C-section. Yeah, you either pass when you're entering, or you passed when you're coming out, or you passed 2 times. Anyway, vagina is a passage for the baby during birth. That's why sometimes you call it birth canal. Guys, don't put feelings here, né? Because this is now science and we need marks. It's not that I'm a bad guy. I'm just at the job. Imagine you go to the doctor, then this doctor is a gynecologist. What do you do? Tells you move the trouser. You say I move the trouser. Move the what? So you are so we will say that this doctor is bad. It's my job. All right. Anyway, what's happening is Las Vegas stays there. All right. We continue to say vulva. Yes, this is the opening. We talked about it. Cervix, we say that this allows, this stretches. Yes, it allows the baby to go through during birth. Yes, when you feel those pains, né? Those pains. Oh, you guys, you must love your mothers. All right. Then we have the endometrium. Endometrium is this, né? Don't know why it's not showing. Yeah, but that's the endometrium. And then this is the ovary. Yes, that's the ovary. Yes. So endometrium basically is where implantation takes place. That's where implantation takes place. Someone is asking me, Z2, ZZ. Vivipara. Oh, you are giving me the other answers. Eh? Yeah, man. Eh? Thank you. Zeze, someone saying my favorite. All right, guys. Thank you very much. By the way, did you know that you are my favorite students? You are my favorite students. Yeah. I'm your favorite teacher. Ne? You are my favorite student. Mm. All right, guys. Let's continue. So basically, that's the structure of a female. Boys, there is no way you can fail this. Huh? You fail a female reproductive system, female reproductive structure. Why? For what reason do you give? Huh. You can't lie to me. But don't forget, guys. Stay what? Stay virgin. Because virginity doesn't lie. And virginity is dignity. It's not opportunity. It's dignity. If you're virgin, that's dignity. If you're not, sorry. If you lost it, start now. If you're married, cool. All right, let's continue. So basically, that's what we need to know about that. The second thing we need to know about is the structure of the ovum. Let me do it here. We saw the structure of the sperm. What about the structure of the ovum? I call it the eye. We use the ovum to see. Use the what? The ovum to see. To see what? To see what you see. You have the outer membrane. Let me draw it nice. You have the outer membrane. Yes. You have the inner membrane. Yes. You have the inside membrane. You understand? Yes. So basically, if you look at it here in our book for distinction material, oh, by the way, I also talked about it. You see, you have the like layer, you have the cytoplasm, and then you have the nucleus. So basically, that's what you need to know about the, yes. So basically, yeah. So the gel-like layer, those are the things which are there. The cytoplasm, that's where, and then inside there, you have the nucleus, and you know the function of the what? Of the nucleus. That's it. That's it. We don't want too much stories there. We don't want too much stories. Yes. So we are still focusing on those 40 minutes. So, yeah. So we are saying that puberty, I'm not going to waste time there. Just here that we'll talk about it again. What is this? Development of the Graafian follicle, ovulation. People, if you are really struggling with human reproduction, here you can't leave this out. I'll show you. If you are really struggling, you can leave it out for now and then get marked somewhere else. But this you have to get it. So you have the development of the the Graafian follicle. Basically, the ovum is inside there. This is mature Graafian follicle, and then this is ovulation place. That's ovulation is taking place. And what happens? Yes. So, just a moment, guys. Yes, got it. Let's label it. Let's label it. Let's label it. First of all, you have, what is this? This is the developing Graafian follicle. What is this? This is a mature ovum. What is this? This is a mature Graafian follicle. What is this? That's the process of ovulation. That is this. This is the ovum has been released. What is this? This is corpus luteum. But the corpus luteum is changing the size. That is an indication that it indicates something. You understand? It indicates something. That indication, it means that the person is not pregnant. Ovum was not fertilized. You understand? So you need to know how to label this. After knowing how to label it, you need to know whether the person is the person became pregnant or the person is not pregnant. You understand? Yes. So you need to know that. All right. So basically, that is it. Puberty, that's not a big deal. You just look at yourself. What things do you have as a guy? What makes you to be a guy? Those are secondary sexual characteristics. And then in females, the same story. What makes you to be the way you are? The only thing I'm going to talk about in puberty, what causes puberty in female is estrogen. That's the hormone called estrogen. And what causes puberty in male is testosterone. We talked about it, secondary sexual characteristics. We talked about it. So now let's do a recap for female reproductive system. Let's do a recap. We said that know how to label. The second name for the vagina is a birth canal. Done with that. Know all the functions of the female reproductive system. Then now know how to label this and the functions. The functions, we can say that we will talk about it more, but the functions, know how to label everything. Know which hormone does Graafian follicle produce. It produces estrogen. Yes. And which hormone does progesterone produce? Sorry, copper sodium. It produces. Yeah. I'm back. So basically, that's it. Oogenesis, spermatogenesis. Guys, you don't have a lot to talk about here. However much those books you are reading, they are talking about a lot. You don't need to talk about anything too much. What is gametogenesis? What you need to know is what is gametogenesis. Gametogenesis is divided into two. That is spermatogenesis and oogenesis. That's what I mean here. The gametes, gamete, from the word gamete. When you say gametogenesis, which means gamete, from the word gametes. So these gametes, you either produce the sperms or you're producing the ovum. You understand? So gametogenesis is divided into spermatogenesis and oogenesis. So when they say formation of gametes, that is gametogenesis. Formation of sperms, that is spermatogenesis. Formation of the ovum, that is oogenesis. That's it. That's it. Spermatogenesis in male, oogenesis in female. How do I know it is? You'll see at the end of the day, you have sperms. So that's spermatogenesis. You understand? Which hormone is responsible for this? Testosterone. You see, because we talked about it. That's why I say that. Please, you need to know about that. Which hormone is responsible for this? Formation of the ovum. Yes. But what hormone is responsible for this? Is the follicle stimulating hormone. You understand? It helps to stimulate this. You understand? To stimulate the development of this small cell, which has to undergo mitosis, so that now this one cell will develop while the other three will undevelop or will remain small, which you call the polar bodies. So, okay, I'm going to explain to you how to write in an exam. So, when you talk about spermatogenesis, guys, yes, you talk about that under the influence of testosterone, the diploid cell in the seminiferous tube. In those tubes we talked about, yes, they undergo meiosis to form 4 haploid sperm cell. Simple. Don't write this spermatid, don't write this spermatozoon, spermatogonium. Don't talk about spermatocyte. We don't talk about nothing. You only talk about under the influence of testosterone. We give you a mark. The diploid cell, the cells with 2N, these ones, give you a mark. They undergo meiosis. Mark to form haploid sperm cell. That's it. Don't stress. Don't waste time. Yes. Then when you go to, don't think about this. The only thing, if they bring this, they can ask you, how many number of chromosomes which are here, what is here. If you see 2n to n, automatically that is mitosis. If you 2n and 1n, that is meiosis. Yes, this is mitosis. And then now, if this is 20, then automatically also this is going to be 20, it's got 2n to n. And then if this is 20, this is 1n, then this is going to be 10. Even this one is going to be 10, it's going to be 10, it's going to be 10, it's going to be 10. That's it. So here, when they bring this diagram, they're just asking you how many chromosomes are there. And they might not tell you that that is a human cell. Yes, it could be something else. So don't think that always we're going to bring a human cell. Yes. And then here, structure of a sperm, we saw it. Yes. Ovum, under the influence of FSH, the diploid cells still undergo, this diploid cell undergo mitosis. This mitosis, yes, form numerous follicle. Only one cell will enlarge to form a mature cell. So meaning that only one cell will enlarge, you understand, to form one mature cell. So it survives. These ones, they degenerate. You understand? And the one which survives is the one which makes to be the ovum. So you say that under the influence of FSH, the diploid cell in the ovary undergoes mitosis. When it undergoes mitosis to form, what does it form? Forms numerous follicles. Cool. One cell inside the follicle enlarges and undergoes meiosis now. Only one cell here will undergo meiosis. It will undergo meiosis 1, then undergo meiosis 2, and then now to produce only one. Yes, among the 4, only one which is produced survives to form a haploid mature ovum. That's why you produce one kid per reproductive cycle. If you produce twins, either the sperm, one sperm fertilize the ovum into 2 equal parts, you understand? Then you have identical twins. That's why you can never find an identical twin which is a boy and a girl. Never. Why? Because it is one sperm cell, one ovum. You understand? Then these other ones, maybe you super ovulate. You produce too many ova, meaning that you can produce more than that. That's when you can produce non-identical twins. That's what it means. So guys, that's it. Don't waste the time stressing too much. That's it. Yeah, just this, nothing else. Yes. So that is clear. So yeah, this is what I was talking about, the menstrual cycle. Here, I know that it's a problem to you, but if you see that this is a problem to you, find a way how you can get some few marks from there. If they bring a graph, get those marks. If they don't bring a graph, they bring something else, still get those marks. We either bring this or we bring this or we bring the graph. You understand? If they don't bring this, that, all these things, yes, it is very important, but you can look at the graph and then you are able to understand. You don't need to know all these things in yes, read them, ne? Read them. Yes, but I'm trying to show you that when you know this graph, automatically you know this, and you know this. Let me show you. If you look at this graph, question they can ask you: did the fertilization take place? If you look at this graph, you have to just look at what shows that fertilization took place. Before we talk about the hormones now, before we talk about what is it, what does it produce, we have not talked about that. Did the fertilization take place? You have 3 things to look at. Did the fertilization take place in these graphs? 3 things to look at. First of all, look at the structure of corpus luteum. That's why I say that you need to know how to label this. I told you that. You need to know how to label that. Look at the structure. If the structure of corpus luteum degenerates, automatically fertilization did not take place. Look at it here. This is Graphian follicle, and then after ovulation, it's corpus luteum. Does it have the same size? No, it degenerates. So fertilization did not take place. And if fertilization did not take place, if corpus luteum degenerates, automatically look at the level of this yellow part, the level of progesterone, because corpus luteum produces progesterone. Check, it drops. So the level of progesterone also drops. Okay, simple. That's done. What about, look here, the endometrium? Did it shrink? Did it bring about blood? Was menstruation accompanied? Yes. Automatically, you can't be fertilized. You can't be pregnant at the same time you are undergoing menstruation. If you see that blood is something else, it's not menstruation. Yes. Yes. So if you have such, if you're a lady, you are outside there, you are pregnant, you can't be pregnant 2 times. Even if it is in the morning, evening, lunch, supper, never. You will never. I'm telling you, you will never. So if you see that blood coming out, just know that it's something else. It could be an infection. Yes, just go to the doctor or go to the pharmacist so that they can help you out. Ne? Don't sit there. We still want that baby, which is the baby who is inside there. Yes, all right. All right, we are saying that did fertilization take place? Think about it. No. Why? Check. Corpus luteum. Did it generate? Yes. Didn't it degenerate? Did not take. It took place. Look at the level of progesterone. Did it go down? Yes. No fertilization. Is it going down? Yes. No fertilization. Is it up? Is it still up like this? Yes. Fertilization took place. Check about the menstruation at the end, not at the beginning, at the end. Yes. If menstruation is not there, is the endometrium shrinking? Starting, it's like a sponge. Is it shrinking? Yes. If it shrinks, fertilization took place. If not, don't think about it. It didn't take place. You understand? So that's how we try to label those questions. Now, another question they ask you, when did the ovulation take place? Ovulation in most cases takes place when you see that spike of a specific hormone spiking. Check this spike, that spike. That's when fertilization, that's when ovulation took place. You see, even there, the ovum is being released. Which hormone spikes out like that? It is luteinizing hormone, LH. When you say LH, we give you a mark. But it's better to write luteinizing hormone. But if it fails, guys, you write LH. Yes. If it fails, write capital L, capital H. We give you a mark. Yes, so it is day 14. Now, what is the function of this? Why is it on day 14? It causes this ovulation take place. You see? And also changes this empty Graafian follicle to corpus luteum. Once this thing has been released, so it changes this empty Graafian follicle, then it puts it to corpus luteum. I think I'm clear there. Eh? You are seeing how we can get marks from this without stressing that much. Because we're not going to ask you questions that describe the menstrual cycle. Those things were there before. Those are gone days. So, basically, I'm not explaining the menstrual cycle because if I'm supposed to explain the menstrual cycle, I'm supposed to explain very well. You understand? Yes. Please know the function. Know the hormone here. Check here. Check here. Check here. Check here. This hormone FSH works here. This hormone LH works here. This hormone works there. This hormone works there. So if it is before day 14, you cannot think about follicle-stimulating hormone. You cannot think about, if it is before day 14, you cannot talk about luteinizing hormone. You cannot talk about progesterone. It must be towards day 14 and after. If it is after day 14, you cannot talk about FSH and estrogen. You can't, because these things, they work before day 14. You see, you just have to know how to manipulate this, and then you are able to obtain that distinction. You understand? Yes. So, that's it. The last part on reproduction, what you need to know about reproduction, so that I give you some questions. Yes, is copulation, that's sex. Different ways how organisms can do sex. Humans, I don't include you because we're not, you're a disaster. What practice everything? Humans. All right, fertilization, you know that we talked about it. Process of fertilization, when they talk about the process of fertilization, you talk about the acrosome approaching, the sperm approaching the ovum, the acrosome fertilizing, releasing the enzymes. The enzymes digest the membrane of the ovum. The haploid nucleus of the sperm fuses with the haploid nucleus of the ovum to form a diploid zygote. Basically, that's what you need to talk about. Just look at, think about the sperm coming, sperm coming, sperm coming, sperm coming. Remember, I told you a moving sperm. Yeah, so you are swimming in the swimming pool. You are going. The ovum is here. Automatically, you won't start with the legs. You start with the head. Once the head comes into contact, think about a mango. Yes, you are going. The mango is coming. You are bringing the mango. Coming to the mango. When you reach the mango, what do? You just mango. You are nice. You open the mouth so that now the acrosome breaks so that the enzyme is released. That enzyme is released on the mango. On the what? On the ovum. You open the mouth so that you can bite. You understand? How they bite? You understand? Yes. So it's the same thing. When the sperm swims. When it reaches there, automatically, once it releases the enzymes. Once the enzymes are released, automatically, what happens? It digests the membrane of ovum. The haplonucleus of the sperm will fuse the haplonucleus of the ovum to form a diploid zygote. That's fertilization. Life science is easy. Right? Why is it easy? If it is difficult, you have to first love it. When you love it, it will love you too. It will love you too. All right. So lastly, I'm talking about this. Make a mistake. Make a mistake, you go to the paper without knowing that. Make a what? Make a mistake. You go to the paper without knowing that. That will be a major or terrible mistake. So here, the sperm fertilizes the ovum. What does it form? It forms a zygote. We call this a zygote. The zygote, look, this zygote will undergo what we call mitosis. You understand? When it undergoes mitosis, it forms, remember mitosis, one cell divides from two cells they divide from 4 cells. So it ends up forming a ball of cells. That ball of cells we call it morula. The morula undergo mitosis repeatedly to form a halo. It has a space. It has a space inside. Form a halo ball of cells, which called blastula. The blastula now forms finger-like outer layer, which called the chorion. Remember, I told you that the outermost membrane, embryonic membrane, is called the chorion. So we call it chorion. It forms the outer layer called the chorion. You understand? Forms what? The outer layer called what? Chorion. That chorion forms finger-like projection. This chorion forms finger-like projection. Yes, those finger-like projection, we call them chorionic villi. That's when maybe you start vomiting in the morning. Yeah, when you are pregnant. Yeah, you are feeling the pain. Why? They are entering you. Yeah, you know how they enter, unless you have never been pregnant. If you have ever, yeah, you know how it feel like. Yeah. So now those finger-like projection. You know, when you are writing these papers, you must start thinking about it if you have ever experienced it. Oh, yeah, finger-like projection. When they become there, then this part is one which becomes the placenta. So how do we mark? After fertilization, this is the one end. When they fertilize, you form the 2N. 2N, it means that this zygote is 2N. It undergoes mitosis. This is also 2N to form a ball of cell. So when you are marking, this is what you are looking for. Let me clear up for you, and then you are able to understand what I'm saying. Let me clear up for you, and then you understand what I'm saying. So what I'm saying that after fertilization, a zygote, we give a mark for that. A zygote undergoes mitosis, we give a mark to form a ball of cell. You saying a ball of cell. Where is it? Ball of cell. This ball of cell, we give you another mark. Yes. And how do you call that ball of cell? Called morula. We give you another mark, which undergo mitosis to form a halo. This is why the halo, this space, we want to see it. Halo ball of cell. How do you call that halo ball of cell? Blastula. We give you another mark, which forms the chorion, the outer layer, which called the chorion. You give you another mark, which forms finger-like projection. Finger-like projection, which we call the chorionic period. We give you another mark. What is the finger-like projection used for? It's used for implantation. We give you another mark. How many marks? 1, 2, 3, 4, 5, 6, 7, 8, 9. So see, we only want maybe 4. Simple. Yeah, guys, reproduction, that's it. The remaining part of reproduction is this one. And this one, let me show you what to do here. That's it. And then you have your 41 marks. Let me show you. They ask you this question. Let me show you. Look at this diagram. Let me make it bigger so that you can see it clearly. Yes. They can ask you, you see now the sperm? This is the uterus. They come like this. This is a sperm. This is sperm. This is the ovum. No. Yes, let me show you. Wrong. That's a sperm. This is a sperm. Yes. But now this is a sperm. It moves. It moves there. It passes there. It moves. It finds there. So this is a corpus luteum. The ovum has been released. The ovum will find with the sperm. So fertilization takes place there. Then now this fertilization. Now this one starts to divide. So this is a zygote. Then now zygote forms two cells. These two cells will undergo mitosis to form a ball of cells, which called the morula. Yes, so it's still, I can maybe count the cells, but it can be a morula, and then this is a supermorula. You can see, yeah, that's morula. Yeah, and then now the morula will see that now the blastula is formed. It formed, but it's starting to implant itself. You see, and then now implantation has taken place there. You see, yes. So basically, I can ask you how many cells, number of chromosomes are here. Is this is 1n, meaning that is 23. And then how do you call this process? Is fertilization. How many chromosomes are in here? It is 2n, which is a zygote, which is 2n, meaning that is 46. And then yes, how do you call this structure which is formed? So we just keep on manipulating questions so that we go off. But you guys, the way how I trust you now, yeah, you are able to get those marks. Guys, what is the function of the placenta? Nutrition. You can talk about all the processes you have in the body. Yes, I talk about nutrition, that is feeding, protection, like the skin, gaseous exchange, that's breathing, excretion, that's when you're going to caca, and then sweating. Or the placenta does everything for the baby. Yeah, endocrine function, that is secretion of hormones. So placenta does everything. The only thing which you guys don't talk about and you're always forgetting is the function of the amniotic fluid. Function of amniotic fluid. You students were always forgetting and confusing this. The function of amniotic fluid is not against the mechanical, it's not providing nutrients, because you see it's a fluid, so you think that it is for nutrition. Shock absorber, prevent dehydration, temperature regulation, yeah, pH, you understand? So when a pregnant lady has maybe something which takes her, how is this lady going to be protected? It's going to be protected by this fluid. Remember, a fluid liquid is incompressible, so it's able to be protected. Yes. So basically, that is it. Let's go to our questions now, guys. Let's go to our questions. Last time we saw that one. Can I give you another one? Yes. So I'm gonna give you two, 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 2 minutes. 2 minutes. 2 minutes. 2 minutes, guys. 2 minutes. 2 minutes to write for me there. We're gonna go through them, but we want to see those who have got it right. 2 minutes. Yeah. 2 minutes, guys. 2 minutes. It's nice to be you, ne? All right. Anyway, that guys, ne? The first one is saying the process of maintaining a constant environment, process of maintaining a constant internal environment. Someone said what? Oh, guys, thank you very much for your answers. At least I see you guys. Yeah, man. Yeah, man. Eh? Yeah, I love it. I love it. I love it. So the process of maintaining a constant internal environment is called homeostasis. That one is called homeostasis. So anyone who said that? Good. I'm seeing people say it. The organelles found in large quantities in the neck region of the sperm cell. You see, we talked about them. Yes, we say that those organelles are called large quantities. We call them mitochondria. See, we say them here. Here. You see, say them here. Yes. They're saying that, so that is done. They're saying that the disease characterized by the degeneration of the brain tissue leading to memory loss. Yes, someone said Alzheimer's disease, correct. So we're going to look at it. Yes, so this one is also covered. Layer of the eye that is richly supplied with blood vessels. That is choroid. Did someone say that? Yeah, someone said it. Thank you. The layer in the eye that is richly, okay, we've done that. The type of development in birds in which young ones are born fully developed, born prepared. So it must start with the word precocial. Did someone say that? Yeah, someone said it. Precocial. Then they're saying that the group of the cell in the pancreas that secrete insulin and glucagon, isolates of Langhans. Someone said that? Yes, someone said it. You guys were good students, and you're gonna make it. I'm telling you. I'm telling you. They're saying the structure in the sperm cell that contain enzyme to dissolve the outer membrane of the ovum. The structure in the sperm cell the structure that contains. You understand? Yes, we said it here. We said it here that this structure, yes, it contains enzyme which digests the membrane of the ovum so that fertilization can take place. Correct, which is acrosome. People are giving me the correct answers. Acrosome. Yeah, I will teach you every day, I think. Hey, because I like people who are always participating. Yes. Then you're saying that a group of cells, we have done that. Structure of a sperm cell that contain enzyme, we've done that. Blood vessel that transport carbon dioxide from the fetus to the placenta. Blood vessel that transport carbon dioxide from the fetus to the placenta. Yes. When you are transporting carbon dioxide from the fetus to the placenta, away from the heart, if it is away, it is an artery. So it is going to become umbilical artery. Hey, did someone say that? Umbilical artery. McFerry. Yes, McFerry got it right. But please check about the spelling, ne? The word artery, ne? Yes, artery. It has an R at the beginning, between A and T, ne? Artery. All right, guys. Yeah. Now, these are terminologies. 10 of them, they will come. Make sure you pass them. Make sure you pass them. Because now I want to go to we are done with this. We are done with this. I want to go to these ones now. So that now we have our 95 plus 8. At least we have 100 and yeah, 5, 8, 103. Yes. At least there we know that we're a little bit safe. Yes. Guys, now this section, before I go to the next topic, what to focus on, this section, this is how we answer it. No, before I tell you what to do, can you answer for me 1 point? Can you answer for me this? I'm giving you 1 minute. 1 minute. Then I will do with you that. 1 minute. Please answer for me that. Answer. Yes, I'm not even going off. I'm waiting for answers. 1.3, one boss. Okay. By the way, guys, don't forget to like and subscribe. Like and subscribe. Kio Kio Lebe Kio ne? Yeah, thank you, members. That small contribution means a lot to us. Ne? I'm seeing a lot of members here. Honsi is also a member. I see a lot of members here. Ne? Thank you very much, guys. Yeah, that small contribution makes our channel to grow and buy data so that we can streamline for you guys. Yeah, that is not too much, ne? Yeah, it's not too much. All right, guys. Ne? Yeah, I'm seeing answers coming. Mobile, Ngoma, Ngema, ne? Ngema, ne? Guys, sorry if I'm pronouncing your name wrongly, ne? Yeah, all right. Guys, I've got what I wanted now. Let's go now. 1.1. Extra-embryonic membrane found in amniotic egg. Extra-embryonic membrane found in amniotic egg. Remember I told you about the membranes? Yes. So, this is the answer is both. The answer is what? Let me just write here. Oh, sorry. You know what I'm cleaning up, candy. The answer is both A and B. That's the answer. If you wrote both, it's wrong. If you wrote A and B, it's wrong. We'll cross it. You understand? Because the instruction is saying write both A and B. Do you see how you lose marks? Yes, that's how you lose marks. Then you're saying that structure in the ear that absorbs excess pressure wave from the cochlea. The structure which absorbs excess pressure, yeah, they're saying pinna and then autonomic. No, they're saying absorb. It's the round window, so it's not here. So the answer is none. You understand? Yes. Then they're saying that the structure that transports semen out of the body. So this one, now you see now, any question which is related to reproduction, you are able to answer it. It's urethra. So the answer is B only. You see, if you wrote B, it's wrong. Yes, it must have the word only. You must have to follow the instructions. You understand? You have to follow the what? The instructions. A only, B only, both A and B, or none. If you change the style, we don't give you anything. You understand? We don't give you anything. Thank you very much, guys. I'm seeing the answers are flowing like water. Ne? Yes, they are flowing like what? Thank you very much, guys. This one, process that produce for mature gamete in human from a single diploid cell. The process that produce for mature gametes. Yes, gametes, they are saying for mature. In this case, we are looking from a single diploid cell. Yes, both of them are producing the gametes, but only sperm, spermatogenesis. You see what I told you about the spermatogenesis? I said that it produces 4 sperm cells. 4 sperm cells. You see, this one is only one, while this one produces 4 of them. This is what they are saying, 4 of them. See? Yes. So that's what they are looking at. So the answer is B only. Yes. They're saying that defense mechanism that protect the plant against herbivore, we're going to look at them, but they're saying defense mechanism, a defense mechanism that protect against herbivore. Both of them are defensive mechanisms, you understand? But herbivore, to eat them is basically, you are talking about the thorns. Yes, so the answer is going to be A only. The nerve that transmits impulse from retina, the nerve that transmits impulse from the retina to the brain is optic nerve. So the answer is A only. So basically, that is it. That's what you're supposed to look at. So yeah. Okay. Production. Let's do this one together. When you talk about what is A, you see now, we talked about it. What is A? A is Z. How do you write it? Look at the first gland is called seminovesical. Check. So it's the first gland, meaning that A is seminovesical. The second gland is Z. Prostate gland, they talked about it. The last gland before you go out, it is the Cowper's gland. See? Yeah. So basically, it's a Cowper's gland. This is prostate gland. This is seminovesical, and then this one is what? This one is the testis. So part A is seminovesical, hormone produced by B, testis obviously. So automatically the answer is going to be what? It's going to be testosterone. Explain one function of the fluid secreted by prostate gland. You see, function produced by the prostate gland. Function is to neutralize the acidic nature of the vagina. Yeah. So that is it. Then we go to yes. Yeah, I will talk about the other one when we reach there. So let's here in a human response to environment. Remember we said that the max you're gonna find there is 54 marks. So if it is 54 marks, 54 marks, guys, 54 marks is a lot. So basically here what you need to know is central nervous system, peripheral nervous system. What's the difference? The central nervous system is made up of the brain, is made up of the what? This brain and the spinal cord. While peripheral nervous system is made up of these other nerves which come from the brain and the spinal cord. That is it. Central nervous system, what is made up of? Peripheral nervous system, what is it made up of? Yes, brain, spinal cord. The peripheral nervous system, we talk about other spinal nerves. So that's the first thing. The second thing you need to know, how is the brain protected? The brain is protected by the cranium. How is the brain protected? It is protected by the cranium, by the meninges, and then by the cerebrospinal fluid. That's it. How is the brain protected? It's by the cranium, meninges, cerebrospinal fluid. How is the spinal cord protected? The spinal cord is protected by the cerebrospinal fluid and the vertebra. So cerebrospinal fluid, that's why it's called the cerebral, which is a part of the brain, and then spinal, which is a part of the spinal cord. Yes. There's a difference between the spinal cord and the spine. Because the spine is this thing you see. So it's better to call it the vertebra. Yes. So from there, you need to know about the brain. What do you need to know about the brain? Labeling. This one is simple. Just label the structure of the brain. The biggest part of the brain is cerebrum. I did this on Study With Me. Yes. You have the hypothalamus. It's simple. What does it do? At least you know two functions of each. Don't go to the paper without knowing two functions of each. Know two functions of each. Please. Yes. So at least know two functions here, two functions here, two functions here, two functions here, two functions here. But what confuses students more is when session, because we know that this cerebrum receives and interprets all the sensation. So now in this case, they don't say all sensation. They say they bring someone is seeing which part of the brain is uh, uh, responsible for that. You talk about seeing is a sensation. Sensation is controlled by cerebrum. You understand? Then number two, what students confuse, they can't differentiate between medulla oblongata, spinal cord, and the pons. Pons. This is the pons. We're no longer asking it. The second swelling is the medulla oblongata. Then just down is the spinal cord. So students don't differentiate between the spinal cord and the medulla oblongata. Remember the function of medulla oblongata. It transmits impulse between the spinal cord and the brain. So it means that it is almost the same area. You understand? Yes. So that is the medulla oblongata. So guys, you need to know the functions and the part, function and the part, function and the part on the brain. Know how the brain is protected. Don't go to the paper without knowing it. Don't say that the brain is protected by the skull. I think I forgot about that. The skull. Don't say the skull. Say protected by the cranium, because I explained that very well, that cranium is different from the skull. The brain box is what called the cranium, but the whole thing here is what called the skull. So how is the brain protected? Is protected by the cranium, not the skull. So some questions can ask you such questions. What is A? A is a spinal cord. B is medulla oblongata. C, E is cerebellum. D is cerebrum. E is corpus callosum. You don't have to know the functions of each. Corpus callosum allows communication between two hemispheres and then joins the two hemispheres together. This one, you have to know them. You have to know them. Yeah, it receives and interprets all the sensation, and then it is a center for higher thought processes. It controls all voluntary actions. So you have to know them. You have not at them. You them. You understand? You cannot go without knowing the brain. How is it protected? And then number two. So that one is done. Peripheral nervous system. What you need to know mostly is the sympathetic and the parasympathetic nervous system, whereby peripheral nervous system, sympathetic and parasympathetic, that's what called the autonomic nervous system, which called the fight or you flight. You either flight or you fight. This is also work together with the adrenaline. These two muscles, they work antagonistically. They work in opposite way. So basically, that's what you need to know. Not that much about this. Is it clear? Yes. What are some of the effects of adrenaline or some of the effects of sympathetic nervous system? We usually ask you this more than this. So it's the same thing. Sympathetic, if you just think about the sympathetic or the effect of adrenaline increases the heart. No, don't even waste time. Yeah, think about the lion comes. What do you do? You open the eyes, the pupil dilates. The heartbeat, how does it beat? It starts to beat fast. You see, if it beats fast, what does it mean? Oh, yeah, you see that those things you do in terms of emergence is what you explain. You understand? Vessels of the skin, they constrict, meaning that less blood flows there. How do you feel? You feel energetic. You want to fight. Ne? Yes. It means that now it's flowing through the, through the brain, and what is flowing through the brain, and what, and the flowing through the brain and the muscles, so that now more oxygen is taken to the brain and think very fast. How am I going to go out? So all those are the, so if you check here, heartbeat increase, the blood vessels of the skin constrict, increase in the blood pressure. You see the relaxation. You end up even urinating yourself because the bladder, you don't know. Yes, the people dilates you. Yeah, someone was asking me a question. Why is it that a person who is in love with someone, the people dilate when you look into the eyes? They dilate, yes, because you are like in an emergence. Yes, you are like you are uncomfortable. Yes, so adrenaline is being released in the body. You understand? So those things you have to know them, you guys, ne? Yes. So basically, that is it. That's what you need to know about it. Then you need to know the structure of a neuron. This one, guys, you need to know them. Structure of a what? Of a neuron. This one. You need to know the structure of a neuron. The dendrite, the cell body, the axon, the myelin sheath. Please know, dendrite transmits impulse towards the cell body. Right now, done with the cell body, the part which transmits impulse away from the cell body, that's the axon. You understand? Yes. The part which speeds up the transmission of impulse, that's the myelin sheath. Yes, what we are talking about there. You see? Someone is saying, is parasympathetic voluntary action? No. Sympathetic and parasympathetic, those are involuntary actions. You do them without your own will. Yes, those are involuntary actions. So, myelin sheath, we saw that. Cell body, that's where the nucleus is found. So, basically, cells activities and then also inheritance. That's genetics. So, you cannot go to the paper without knowing two cells. That is the sensory neuron and, sorry, the motor neuron and the sensory neuron. This one you don't usually ask you. And what's the difference? Here, the cell body is along the neuron, the cell body is at the terminal. You see? So, you tell us the at least two differences between the two. Know how to draw A and B. Know how to draw that. Please don't go the paper without knowing how to draw that. Eh? Yes. So, three neurons, sensory neuron, interneuron, and then motor neuron. This and this, they belong to peripheral nervous system. This one belongs to central nervous system because it's inside the central, is a part of, an integral part of what? Central nervous system. Central nervous system. So basically, please don't go to the paper without knowing how to draw this structure and this structure. Make sure that you know how to differentiate the two. Reflex arc. You can leave the rest, but you can't leave the reflex arc. Don't make a mistake. Questions. I told you when I was even on study with me. Reflex arc. What is a reflex arc? Path traveled by the impulse during the reflex action. What is a reflex action? It's a rapid automatic response to the stimulus. Examples of reflex action: blinking of the eye, knee jerk, touching, withdrawal of the hand from the hot object. These things you must be singing them. What is the function of the reflex action? Is to protect the body against further harm. Yes, maybe it can be harmed, but it protects the body from further harm or from danger. Yeah, because you blink the eye so that that thing which is coming is dangerous, and you do it without your will. That's why you say that it's a rapid, automatic, or it's a rapid, fast, involuntary action. You see, yes, you have to be able to answer that, and it's always negative. You cannot say that, oh, this hot thing is nice. No, you can't do that. When you touch the hot object, whether it's a baby or what, the answer negative. You withdraw. When you step on a sharp object, the answer is negative, remove the leg. When the stone come into your eyes, you can't say, oh no, the stone is coming. Yeah, no. It's negative. So meaning that its instance is to protect your body from that danger which is coming. That's why we're saying that from further harm or from danger. Mm. So, guys, I think I'm clear there. So you need to know how to. Then what you need to know about the reflex action also. What will happen if the sensory neuron is damaged? The motor neuron is working. What will happen is if the motor neuron is damaged? The sensory neuron is working. What is the function of? So all those things. Organize a simple question here for you. Yes, here is the reflex arc. So you have to know how to label. This is a we talked about it last time. This is this it's a sensor because this is the receptor. So automatically, you guys don't forget about my handwriting. Leave alone. Um, receptor. Yes, that's the receptor. And then now this is the sensory neuron. Sensory neuron. And then now it enters here when it comes like this. Yes. So this is the ventral root, the dorsal root G. Let me first clear it up. This is the dorsal root. It's the dorsal root. And then now this swelling is called a ganglion, dorsal ganglion. Then now you have here this one is the interneuron. Then you have the junction here and the junction here. Let me clean it up so that you're able to see. There's a junction where this neuron joins this and this neuron joins this. If you look at this book, you can see the junction here. Yeah, sometimes we call it the synaptic, yeah, the synaptic gap. Yeah, synaptic gap. In real science, we call it synaptic cleft. So, but here in your level, we call it synaptic gap. So it's to neuron. Its name is a synapse. So synaptic cleft is a part of a synapse. But now we call it that synaptic, that space. Yes, we call it, that's where the two neurons join. So we call it a what? A synapse. So that's a synapse. Also, that's a synapse. This is the ganglion I'm talking about because this the middle, the cell body is in the middle. So it means that this is a sensory neuron, and then this is the motor neuron. So basically, that is it. And then now you have to label this. So this is the connector neuron, and then this is the spinal cord. That is the synapse. You see, so you have to know how to label this, guys. Please don't go to the paper without knowing how to label. Then lastly, on this, what will happen? What will happen if sensory neuron is damaged, but the motor neuron is working? If the sensory neuron is damaged, the person will not be able to feel because it is damaged, but you will be able to respond. And if the motor neuron is damaged, the person will not be able to feel. No, the person will be able to feel, but won't be able to respond. That's how you're supposed to the stimulus. That's how you're supposed to answer those questions. If the motor is the one which is damaged, you can't send the information to the muscles. You understand? So it means that you will be able to feel the pain, but you won't be able to respond. You understand? Muscle pull. Yes, you are feeling the pain, but you can't do the leg. And then your sensory is damaged, you won't be able to feel, but you'll be able to respond. I told you, removing a tooth, they inject you with the chemicals there. You are able to, when they ask you to that open the mouth, you're able to open the mouth, but when they are removing the tooth, you can't feel the pain. You see, so you are able to respond, but you are not feeling. That's what is happening. Label, so we have labeled them microscopic gap. You see, microscopic gap. This one, that's gap which you call the synapse. And then you need to know the function of the synapse. We say that allows impulse to move in one direction. That is the major function of the synapse. Number two, it prevents continuous stimulation of the organs or structures. Yes, because it keeps on stimulating. So explain one consequence if A is damaged. It's the same thing which we talked about. A is a sensory neuron. Why? Because you see it has a swelling here, so it's a sensory neuron. So it means that you will be able to respond, but you won't be able to feel because it is damaged. You can't feel. The impulse cannot move from here. So you'll be able to feel. You won't be able to feel, but you'll be able to respond. Give two examples of reflex action. We talked about it. Knee jerk, bring of the eye, withdraw of the hand from the hot object, all that. So please don't go to the paper without knowing this structure. Draw the labeled diagram of neuron B. You come out here and then you find out what is neuron B. You see that is the motor neuron. So that's why I told you that, guys, don't go to the paper without knowing how to draw A and B. You see? Yes. Then that is the reflex arc. How do you explain the transmission? Yes, you explain transmission from here, the impulse enters here, you talk about how it moves. That is what is being written here. But as long as you know the other one, at least you know some marks. There is no way how you're gonna fail. The significance of the reflex action we talked about allows impulse, yes, to move very quickly and protect the possible danger. You understand? For injury. So we talked about it. I said this is a synapse, but know the function of the synapse. The junction, what is a synapse? Is the junction between, but the major function allow impulse to move in one direction and also prevent continuous stimulation of the neuron. So guys, I think I'm clear. You see even there, if you check the questions, you'll be able to understand. If they bring it like this, still you're able to answer it. Why? Because this is the cell body. It's along the neuron. Definitely this is the sensory neuron. And then this is the cell body is at the terminal. So it means that the impulse are moving in that direction. See? Yes. So that is Alzheimer's disease. We talked about them even in the terminologies. Alzheimer's disease, that's when you are always forgetting everything. And then multiple sclerosis, and it means that your memory is being destroyed. While multiple sclerosis, the myelin sheath, this outside part of the is being damaged. You understand? So we talked about this, guys. I think that now you are getting it there. The eye. What are you supposed to focus on? Two things. If you want to get a distinction, you cannot go to the paper without knowing two things. I'm not going to go through them. Yes. Know the function of the part of the eye. Know the functions of the eye. Yes. If you don't have, there is a very a simple video, 70 minutes. Yes, watch it. I explained everything. Know the function of the eye. Then know pupillary mechanism and accommodation. This one. Accommodation. Accommodation, you cannot go to the paper without knowing accommodation. When you're memorizing, label, answer only one. If you know the nearby object is the opposite for far object, and know that it's only nearby object if it is less than 6 meters. It is a far object more than 6 meters. You understand? So a nearby object, 6 meters. Far object, more than 6 meters. I think guys, you need to know that. Memorize one. How do I know it is about accommodation? When are they talking about accommodation? When they talk about the lens, please don't say pupillary mechanism. When they talk about the lens, the answer there is accommodation. When they talk about suspensory ligaments, when they talk about this thing, that is accommodation. When you are answering, you must answer based on suspensory ligament, ciliary muscles, lens. Other things, no. When they talk about iris, this other one now, when they talk about this, yeah, when they talk about this, yes, and discuss space, that space, then that's when you talk about pupillary mechanism. I think guys clear there, né? Yes. You must talk about now pupillary mechanism. But if they talk about the lens, please, guys, there's nothing I'm gonna change there. Ciliary muscles contract, suspensor ligament slacken. Guys, don't use relax. Use the word slacken. Slackens. It does slackens. It doesn't what? Doesn't relax. And then now the tension on the lens decreases. It means that there was too much pressure on the lens, but now it decreases. Now the lens becomes big, and then the more light is reflected to the retina, or reflected more to the, then the clear image is focused onto the retina. So that's it. That's it. Yeah. Mm. So near object, distant object. That one is simple. Pupillary mechanism is a simple thing. We have two muscles, two conditions. We have radial muscles, radial muscles, radial muscles, relax. Sacral muscles, contract. So radial, we have two muscles. We have the radial muscles, we have the sacral muscles. We have relax, we have contract. When when it is dim, you mix things up. When it's bright, you don't mix things up. So in a bright light, we don't mix them up. So R is gonna go with R and the C is gonna go with the C. Radial muscles, relax. Sacral muscles, contract. When you look into the light like this or the sun, what do? Do you put those things? Do you put those things? The eyes open like this when you're looking to the sun? No, you close the eyes. When you close the eyes, what happens? It means that the pupil constrict. So in the bright light, the pupil constrict. See, now you know that. So radial muscles relax, sacral muscles contract. The pupil, diameter of the pupil constrict. If you constrict the pupil, this light will enter the eye. That's it. Yes. So guys, that's it. Yeah. So guys, that's what you're supposed to know. So make sure that you know the dim light and the bright light. Is it clear? Yes. Then we go to defect. Eye defect. If you are short, you are able to see near object. If you are short sight, you are able to see near object. If you are far object, you are able to see far objects. You understand? Yes. So, but what causes? If you see the image is focused, check. If you are near, how do we bring these questions? We just bring a small scenario and we ask you 1 or 2 marks. So if you are short sight, it means that you are able to see objects which are near. If you are long, it means that you're able to see objects which are far. How do I know that this is short sight? What causes that? Eyeball too long. It means that the image is formed before, because the eyeball is too long. You can't jump through the what? Just jumping the river. You can jump the small maybe stream, but the river is too big, so you will end up not making a full jump, like to the other side of the bank. That's right. Yes. So that's the same thing. So the image is focused. The eyeball is too long. The image is from before. And then how do we correct that? We have to make a diverging lens, which we call the concave lens. Then long sight is you able to see near far object, but you won't be able to see near object. Make sure that long sight you are able to see. What causes that? It caused by the eyeball is too short. The eyeball is what? Is too short. If it's too short, it means that the image is focused onto the retina. behind the retina. You want to be able, the image is focused. So you have to bring it back. When you bring it back, you have to use a convexy, and so that when the light comes like this, it bends it. While here, the image is formed at where? Here. So it's too long. You understand? So it means that you have to put a diverging lens here, so that when the light comes, it diverges it a bit, so that now the image is focused here. So basically, that is it. Astigmatism uneven. This is very common in older people. And the treatment, contact lenses. Yes. Yes. You form multiple images. For example, you have a mirror, break it, you're gonna form very many images. That's what happens with astigmatism. Cataracts, the lens crystallizes. Yes. How do you treat it? Surgery. You go for surgery. Yes. So basically, those are the eye defects. They are simple, very common to they like cataracts more. They like cataracts, guys. Cataracts, they like cataracts. Make sure that at least you know you go when you know that. Here is some question you can practice. Make sure you practice them if you have time. Explain the effect of the person's if E is cut. Where is E? What happens if you cut the optic nerve? Automatically, the impulse won't be sent to the retina. It's not retina, the brain. Part of the brain, cerebrum, because this is a sensation, meaning that impulse won't be able to, and then it means that you won't be able to see. Yes. Part D can be damaged very bright. Part D, which part D? Yes, the retina can be damaged by very bright light. Describe how part A helps to protect part D in a very bright light. You see, now you are explaining bright light. Remember, bright light, don't mix things up. So you're gonna go R with R, C with C. Yes. So it can do this. Yeah. So it contracts, and then now so that less light will enter the eye. Remember, the diameter of the pupil will constrict. You see, and then now less light will enter the eye. Less light. LL. Less light will enter the eye. So basically, that is it. In a condition called presbyopia, lenses lose their elasticity and therefore maintain a constant flat shape. Explain how the condition would affect the person's vision. It's a constant flat shape. If it is a constant flat shape, it means that if it is like that, it won't be able to see. You'll be able to see a person maybe who is far, and then you won't be able to see a person who is near. It means that it won't be able to adjust from far to near or near to far. You understand? So you explain. Suggest the shape. It's not exactly which is coming, because this is in the distinction material book, but you have to know how to apply these questions. Suggest the shape of the lens that may be prescribed a doctor to correct this disorder. So it means that now you are able to see objects which are far, but you won't be able to see objects which are near. So automatically you have what is happening. It means that you are long sight, and the long sight, we say that what you do, you put on the convex lens. So that's what they are trying to give you. And so when they bring the questions on the eye, people they tend to be too difficult, but no pure mechanism, no accommodation. Please don't know the functions of the eye. Sorted. Know the reflex arc. You understand? Know the reflex arc. Know the true structure of the neuron. Know the brain. Know how brain is protected. What is the function of the synapse? What is the reflex action? What is the reflex action? What is the examples of reflex action? Function of the synapse. You talk about what will happen if this is damaged. This is working. The neuron, the sensory is damaged. This the motor is damaged. So simple thing. You'll be able to get distinction. I'm telling you. I'm telling you. Yes, I have less than 30 minutes to finish everything. I mean, here, simple. Label the part, function. Label the part, function. Hearing process, label part A, give the function. Level part B, give the function. Level part C, give the function. Level part D, like that, until you reach to the brain. Level the part A, function. Level part B, like the way how it moves, like this. Hearing is done. Simple. Hearing is done. The only thing which disturbs students here is balancing. Balancing is the problem with the students. Yeah. So balance is the problem here, but here, which organelle, which receptor are found in the cochlea? Organ of Corti. Which receptors are found in the semicircular canal? Is the maculae and cristae. Those are the two organelles. Those are two receptors which are found in there. You understand? Yes. So the only problem where you find the questions which come here, let me say it. You cannot go to the paper. Let me even zoom in. You cannot go to the paper without knowing these parts. The function of that G and the part. These two windows, most especially the round window. You can't go to the paper without knowing that. You can't go to the paper without knowing this part. Yes. Most cause. What is it for? Amplification of sound. You can't go to the paper without knowing that. Yes, we like it. Oval window, round window is absorb the excess pressure or releases excess pressure from the inner. Even the question is asked there. Yes, was it? Yeah, it was asked. It was asked. Yeah, it was asked. I don't remember where, but we talked about it. So this is also another question for the yeah, this is what I've described. Yes. I don't remember where I saw it. Okay. Then they are saying that balance, when you talk about balance, is a simple thing. You need to know the stimulus, you need to know the receptor, you need to know the location. Speed and direction controlled by the cristae. Maculae found in the ampulla. Position of the head is maculae, found in the circulus and auriculus. So what do you say? You say changing the speed and stimulates. Changing the speed and direction of the head stimulates the cristae found in the ampulla of the semicircle. Change in the position of the head stimulates the maculae found in the circulus and auriculus of the semicircular canal. The stimuli is converted into enough impulse which move along the auditory nerve to cerebellum for interpretation. Classic. We don't want a lot. Change in speed and direction of the head stimulates the cristae found in the ampulla of the semicircular canal, Change in the position of the head stimulates the maculae found in the circulus and the auriculus of the semicircular canal, The stimulus is converted into enough impulse which is sent along the which is sent to cerebellum for interpretation to restore balance. You understand? That's what you are looking for, nothing else. Yes. So the defects of the eye, of the ear, is also simple. How do you do these defects? Defects of the ear, yes, is middle ear infection. How do you treat it? Is excess fluid generated in the ear? How do you treat that? You treat that by using the grommets. Number two, because it's caused by the bacteria by application of antibiotics. Nothing else. Nothing magical there. That's it. Yes. Accumulation of wax is just by removal of it. Just remove it, the wax. Then you're able to hear again. If the cochlea is the one which is damaged, then you can use cochlear implant. You understand? If the what? The bones are the one which are solidified, you can use the hearing aids. So no big deal, no what, guys. You have to be flowing. You have to be flowing in the paper. There is no reason why you should not be flowing when it comes to the life science paper. You must be flowing. So guys, if you check it, it means that uh, you are able to get those 54 marks. Don't forget the brain. How is it protected? The functional parts. Don't forget the structure of the neuron, and then don't forget the reflex arc. All the functional parts. What is a reflex action? What is a reflex arc? What is so you see? Don't forget the synapse, the function of the synapse, examples of reflex action. Don't forget the brain, sorry, the eye, the parts of the eye, and then now the mechanism. Memorize one. The mechanism, that is dim and bright light. Memorize bright light is the opposite for dim light. R with R, C with the C. That is bright light. The diameter of the pupil constrict, less light enters the eye. Then don't forget the accommodation, whereby near object and far object. Near object when it is less than 6 meters. When it's far, it's more than meters. You understand? Yes, please don't forget that. Then when you come to the eye defects, short sight means you are able to see objects which are near. Long sight, you're able to see objects which are far. And then astigmatism, it means that you are seeing uneven. You are forming really like these old people. They're just seeing they are just seeing an object moving, but by not recognize the face because of the multiple images which are formed, like your mirror when you break it. Then cataracts, it is as a result of the crystallization of the lens. You see the cloudy substance in there. The lens has become cloudy. How do you treat that? By using surgery. Then when you go to the ear, name the parts and function. Hearing process, you name the part and function in the chronological order, in their order. Hearing process is done. Then we go to balance. When you come to balance, you have two receptors. The first receptor is for position of the head, and then another receptor is change in speed and direction. So change in speed and direction stimulates the crystal found where in the ampulla of the semicircular canal. Change in the position of the head will stimulate the maculae found in the utriculus and the circulus of the semicircular canal. The stimuli is now converted into a nerve impulse which move along the auditory nerve to cerebellum for interpretation to restore balance. Balance is done. You go to what? To the defect, the middle ear infection. When we talk about the middle ear infection, what causes the middle ear infection is the bacteria, accumulation of fluid in the middle ear as a result of the bacterial infection. So how do you treat it? We treat bacteria with antibiotics. Simple. Number two, you insert the grommet so that it can drain that pus in the ear. That's it. Please don't forget the function of the rounding window, the mutation tube. Please don't forget about that. Don't forget about the oscos. Then when you continue, then you go. Yes, the balance is done. So even that is done. So once you finish that, I'm telling you, perfect, guys. You'll be able to get a distinction. I'm telling you. Yes, I'm telling you. So when we come back to our thing, now we have 103 marks secured. Now we are going to this other part. So we are done with that. We are done with that. We are done with that. So now, how are we going? This one is and then this one is big. This one is, but the reason why this is big basically is around 15 and something, maybe, and the maybe 17 something there. You understand? Yes. Why? Because these are two topics. Yes, two topics in one. So let's just highlight them and then we conclude it as a day. Endocrine, you need to know the glands and the function. Yes, you need to know what is endocrine system, what is the function. Yes, you need to know the gland and function. I'm not going to go through these ones, but you have to know the gland, the thyroid gland, the hypothalamus, gland function. Then now you need to know, here you have to memorize one, how the negative feedback mechanism work. Here, guys, I told you that what you do is you have mm, you have you have uh, you have that, and then you have, if this is low, just a moment, if this is high, yeah, so no, okay, let me just draw it again here. If it goes up, this one must go down, this goes down, this must go up. So here you have two statements to talk, but here you have two statements to talk. Normal level of whatever. So in this case, we can talk about anything here. We can talk about anything. Anything is fine. Okay, we have insulin here. Normal level of glucose in the blood. You see that? The first part is increased, the last part is decreased. Two things you need to put here. That's what I'm saying here. Two things you need to put here. All of them, they are like that. You need to know the gland. The gland, you have to talk about the gland, and then you need to know which hormone does it produce. You get there, the hormone you get there, you need to know the function of that hormone, and automatically now it will link to the cycle. So pancreas secrete insulin because I have too much glucose. I need to make it down. So pancreas is the gland, insulin is the hormone. So you go the mark. Now you tell us the function of insulin. Insulin stimulates the conversion of glucose to glycogen. Stimulates the conversion of glucose to glycogen. And then now what happens? The level of glucose goes decrease and then it goes back to normal. So the negative. Um, so guys, I think I'm clear there. You can't go to the paper without knowing that of carbon dioxide. Why? Because carbon dioxide is a little bit different. So what is, we don't want carbon dioxide in the body. So what do is, when increase carbon dioxide level in the body, decrease the pH, because carbon dioxide reacts with water to form that carbonic acid, that acid. So that acid is being detected by the receptors. So an impulse is sent to the medulla oblongata. Medulla oblongata will send the impulse to the heart. So now you start breathing too much. And then the depth of the breath is too much. You understand? And then now the heartbeat will have to pump too much so that it takes carbon dioxide, takes blood takes carbon dioxide from the muscles to the lungs and expelled, and oxygen is picked up from the lungs to the muscles so that more energy is being produced. And then now the level of carbon dioxide decreases and then it goes back to normal. So please don't go without knowing the negative feedback mechanism of the negative feedback mechanism of the I was saying that please don't go the paper without knowing the negative feedback mechanism of carbon dioxide. So what happens? The isolates of Langhans are not working. We call the condition is called diabetes mellitus. Don't say diabetes. Diabetes of what? Diabetes mellitus. Sugar diabetes. Is it clear? Yes. Called the sugar diabetes. Uh-huh. So I think that is clear. So guys, that is it about that. That is it about that. Hmm. And then now, lastly, uh, lastly, lastly, eh, is about the what happens if you don't have enough sorry, you don't have enough iodine. Iodine, if you don't have enough iodine, you suffer from a condition called goiter. So yeah, meaning that if the goit is the thyroid gland is over secreting, so we call it we call it hyperthyroidism. Yes. And then if it is not, it's under secreting, then we call it hypothyroidism. Yes. So basically that is it. What you need to know about that. Lastly, you need to talk about thermoregulation. Thermoregulation, what are you supposed to know? You need to know what you call the sweat. You need to know what you call the sweat. So if you look at the skin, let me just go like this so that you are able to understand. I have the sweat gland, then have the and then I have the radical province. So basically, then I have, let me just draw this the other side so that there is something which I need you to understand. Yeah, this is sweat gland, this is sweat duct, this is sweat pore, and then what produced here is a sweat. So when the blood vessel is big like this and this one is small, it means that now the blood is flowing through or near the skin. So it means that more heat is lost. So this is the hot day. And then now if it is passing here and then it goes back, it means that it's cold. So it won't even see those blood vessels up there. You understand? So this is cold, cold, cold day. Understand? But while this one is hot, hot day. And then now the hot day, if it's like this, it's linked with the sweat. That's why here in the booklet you see that during hot day you have the sweat, you have you see the shunt is too small, so more blood is passing through the skin. You understand? Yes, it's passing through the what? Through the skin. And then it means that more sweat is produced. That's why you're able to see it here. And then it means that the sweat gland is becoming more active. During hot day, hypothalamus is stimulated, then it's impulse to the blood vessel. The blood vessels they dilate, they become wider. This is called vasodilation. And then more blood will flow to the skin, and then the heat, more heat is lost. You understand? Yes, if more heat is lost, it doesn't stop there. More blood is sent to the sweat gland. The sweat gland becomes more active. Then, if it becomes more active, then the sweat gland becomes more active and then it produces more sweat. And then its sweat, well, as it's going, evaporates by cooling of that body. So, someone is asking me, sir, are you gonna do revision and live before the 17th? Okay, yeah, if you want to do the revision, I can do the revision, but it's just a revision. Yeah, just to revise with you. If you have any challenge, you find it. Yeah, we will see how yeah, I'll organize for you guys, and then you see that maybe we can do that revision together. Yes, no problem. We can see depending on my schedule, but yeah, we will see if I'm able to to accommodate you guys in that regard. Yeah, So yeah, I'll try to censor, so it's fine. I'll try to see that I do what? I accommodate you. Yes. All right. So we are saying that another thing now we have talked about that. No, so many questions. How, if you look at this, how do I know that this is the what is the, you have A and B. Here you see the blood vessels are too small, it means that it's a cold day. And then you have blood vessels are big, it means that it's hot day. It means that more blood is flowing near the skin, therefore heat is being lost. You have even sweat. That sweat, it shows that, yeah, you guys were able to remove that. As the sweat is going out, then the heat is being released from the what? From the body. So that is it. Just a moment. Yeah. Guys, someone is asking me, please, do grade. Someone is asking me to say, please, do grade 11. All right, I'll do grade 11, guys, but not today. Also, all right. So we are saying that tropism, this is just small. So guys, that's what you're supposed to focus on. That, don't forget to know the glands and the function, know how the negative feedback mechanism work. When they say the negative feedback mechanism, keeping internal environment constant, then that's negative feedback, that is homeostasis. The mechanism of bringing this to back to normal, that's what we call the negative feedback mechanism. But the process of maintaining this constant environment constant, then that's what we call homeostasis. So homeostasis is brought about by the negative feedback mechanism. I think I'm clear there. So guys, now it means that we have these marks. But I know that yes, it could be challenging for you, but maybe I'll organize for you last eye. I'll organize for you guys last eye. On the 17th I'll come back, and then let me check and see. 17th, yes, 17th is paper 1. Yes, it means that I will be able, yeah, it's a Friday, yes, it's fine. I'll see how I can help you out, guys, so that you're able to score high. Then saying that one saying grade 11 program was also a program. Okay, grade 11. Your voice is being is hard. Your voice is hard, so we will organize grade 11. But be specific, which topic do you want me to talk about, and you want a scope. Yeah, if you want a scope, you let me know, or if you want yes. All right, lastly guys, let's talk about how we get this remaining marks, the 90, the 13 marks. This one is simple. First of all, there is one question I've not talked about. And that is the question which is there in paper 1 and is also there in paper 2. We don't mess up with that question. You understand? We don't mess up with that question because it is there in paper 1 and also there in paper 2. We don't what? We do not mess up with that question. Let me just finish up this. So when you go to tropism, what do you need to look at? Tropism is simple. Know the 3 hormones: gibberellins, abscisic acid, and auxins. Auxins are growth promoter. These ones they work very well in summer, while abscisic acid work very well in winter. So you just need to know that. Then number two, auxins are growth promoter. So you need to know how to apply them here. Please don't go to the paper without knowing this. The phototropism, the auxin effect on phototropism. Don't go to the paper without knowing the auxin in geotropism. You see, either that or that. Lastly, you need to know that this how does this one affect? This is just 1 mark. How does it affect? It prevents the effect of geotropism, because it has to rotate, keeps on rotating, meaning that the light, if light is coming from this side, that light is exposed, so it keeps on rotating and rotating. And if it rotates, it means that each side at the end of the day will have the same amount of light. Yes, that's the what you need to know. If they bring the first layer, this is, you write the word ID, which means X and Y. This must go on the X axis, this must go on the Y axis. So basically this is independent variable, this is dependent variable. So when you are drawing, it means that the years are supposed to go here. 2014, 2015, because this is ID, meaning independent variable, dependent variable. Dependent variable go on X axis, dependent variable go on Y axis. I think I'm clear there. Then you need to know what is if they give you a question, what are you supposed to look at? Basically, you have to look at independent variable. Sometimes you have to look at what is given here. You say that this is the effect of sunlight on, you see, the effect of sunlight on growth. So it means that sunlight becomes the effect, becomes the independent, and the growth becomes the what? The dependent. So growth will be depending on amount of sunlight being received. Then how do we calculate percentage increase? Percentage increase is calculated by final minus initial over initial. Please don't go there without knowing it. Yes, it equals to percentage increase. Then how do we calculate the bar graph? The bar graph is supposed to be having equal bars. The bars are supposed to be the same size. You understand? They're supposed to be the same size. Supposed to be the same size. So if you make the bars different sizes, then automatically you lose.